cruzi, similar to other species of the Triatomine family, even males and females have wings, but in Mepraya species, only males would have wings. The females are ever wingless, okay? This probably is apomorphy in this group, yeah? And in males, there are polymorphisms for wings, yeah? In this species, Mepraya gajardoi, that I described with other colleagues in Chile, the males ever have or are brachypteros, yeah? Similar to Mepraya parapatrica that we described recently. The males ever have wings, but in Mepraya spinolae, the males are males with winged and wingless, okay? The wingless condition in females and males is a neoteny, yeah? And the genes for the development of wings and the thorax, the development are located in Y chromosome. In this species, also there are differences in the morphology of the eggs. Yeah? Then we think that's a very good species. Yeah? How is the distribution, geographical distribution, this species in Chile? Gajardoi distributed in the north in Chile in a coastal line, yeah? Similar to Parapatrica. And the trophic niches is seabirds in the north of Chile. Gajardoi is not a very good transmitter vector of Tricruci, but probably why this parasite birds, and in the birds no development the Tricruci. Spinolae introduced to the valley of the north and central zone of Chile and changed the trophic niche. They parasite preferably to small mammals, rodents, and others. When we realized a reciprocal cross in this species, in Mepraya spinolae, give different results. When you cross winged males with this female that are ever wingless, called in the F1, you have winged and wingless male. But when you cross wingless male with females, ever the F1 is wingless male. The gene is located to Y chromosome. How is the cytogenetic this species? Yeah? The three species have the diploid number of chromosome is 20 chromosome more X1, X2, X2, and I chromosome. In Parapartica, este es Mepraya spinola, that is different in the heterochromatin, constitutive heterochromatin. The autosoma of this species have not heterochromatin, but in Parapatrica, all the telomeres have heterochromatin. Yeah? Then the heterochromatinization is very important for the speciation of Mepraya parapatrica. Okay? The diploid number of chromosomes is similar. The Y chromosome is biggest than the X chromosome. Okay? In the other species, Spinolae, probably in the evolution break the Y chromosome and produce two Y1 and Y2 chromosome. And there is the wingless condition in the males. Okay? This is the habitat, wild habitat, domestic habitat, and peridomestic habitat in this species. This is a wild habitat in the north of Chile. Yeah? In the slide you can see domestic, peridomestic habitat. Okay? In this slide, I show the number of wingless and wingless males or females of Spinolae from collected in wild and peridomestic environments. You can see that in a wild environment, the wingless males is higher than the winged males. Yeah? But in the peridomestic habitat, this is different. The number of wingless and wingless is similar. But when you compare the proportion of wild habitat with peridomestic habitat, there are very significant difference, statistical difference. Here is this result. When you compare the proportion of winged males and wingless male in Colina, until that are wild environment, the difference is not significant. Similar when you compare peridomestic habitat. There are not statistical significance. And the proportion of males of females in this different population is 1 to 1, similar. In this slide, I show the life cycle stage of Spinolae inside domestic environment, in rural house, yeah? In localities of north of Chile. Yeah? You can see that the winged males are higher in a domestic and house, inside house. We never collect wingless males inside house. I never collect eggs inside house. Then the winged males are important in the transmission this disease inside house. Yeah? And probably is the dispersive form this insect in house. The eggs only we collect in wild condition, yeah? In wild habitat. This is nest of rodents in the central zone of Chile, and we collect eggs. How is the infection of Spinolae with the cruci in wild and peridomestic environment? If you compare peridomestic and wild, the positive or infected is higher in wild condition, and is low in peridomestic condition. What happened when you release it or estimate the number of individuals of spinolae infected, positive, and uninfected with the cruci collected in domestic, peridomestic, and wild environments in the north of Chile. You can see, the red bar. In domestic, the uninfected is higher than in peridomestic and wild. And the other result important in this slide is the winged smile. There are not winged smiles inside the house. This is the statistical result when you compare the proportion of positive winged smile with negative winged smile. There are statistical significant. Okay? The conclusion. The winged smiles are more frequent in wild population than in domestic and peridomestic environments. In South House, winged smiles are significantly more frequent than winged smile female and nymph. X was found only in wild population. This suggests that the reproduction of spinolae occur preferably in wild conditions. The principal invasor toward domestic environment, there are the winged smiles. This factor suggests that the dispersive form of this species is similar to other species of insects with winged and winged smile. Feeding deprivation could be explained the low infestation of winged smile collected inside house in comparison with these winged smiles in wild habitats. An insect has been postulated an ecogeographical theory of thermoregulation according to this theory, the insect with longer wing would be distributed in colder climates. Those without wing or very short wings could be adapted better in warmer climates. In base to this hypothesis, the wing of winged males of spinolae could cause greenhouse effect, and heat could accumulate in their body, and they could seek refuge inside the house where there are a nice environment. Conversely, winged males disperse the heat body more easily and they will be better adapted to wild hot area. This we call the refuge theory. Then in spinolae could be a disruptive selection. In domestic and peridomestic environments, the most probable cross is winged with females, and in wild environments, wingless with the females. Okay. Other research that participate in this research are Cristian González Aravena, Carolina Reyes Valenzuela, the Institute of Entomology, Metropolitan University of National Educational Science, and also Public Health Institute, Ministry of Health of Chile, Daniela Blanco Carvalho, Faculty of Pharmaceutical Science, State University of Sao Paulo, Brazil. Thank you very much. And excuse me for my English. And I solicit that if something here that speak Spanish, help me in the translation in your question, okay? So.
very much. Session is open for questions. Yes, please.
Thank you for this.
It's on.
Thank you for this nice presentation. I just was wondering, is it only the male mites that spread this disease, or do females also have something to do with that?
The Chagas disease?
No, I mean the male mites. You only talk about the male in this research, and I was wondering if these female mites also are vectors for this trypanosoma disease.
Ah, yes. We released banding for this chromosome, and diploid number is similar in autosoma, but it's very different in sexual chromosome.
Oh, okay.
Two of these species have 20 autosomes, and X1 and X2 and Y chromosome. Mepraya parapatrica and Mepraya gajardoi. But in Mepraya spinolae, there are a polymorphism for Y chromosome. There are neo-E chromosome. And in one disease, Y chromosome is the gene for wings. And the other neo-E Y chromosome, there are not the wing for the gene for wing. Then this situation is the polymorphism. Explain the polymorphism for wing in spinolae. Then there is wingless male that have in the Y chromosome the gene for wings. In the other neo-E Y chromosome, there are not the gene for wing. Okay.
Oh, okay.
Then for us, the wingless condition is una apomorphism. Okay.
Okay. Thank you.
In the neoteny, there are problems in the development of thorax and also in the wings. Okay?
Okay.
But the females are fertile and they reproduce. Oviposit eggs and are fertile. Okay?
Yeah. Okay. Thank you.
Well, over there.
That's a very nice presentation. I want to ask about is different kind of red-winged bug. Did they go to feed different time or not? They go at night, like a mosquitoes. They have peak of go to hunt or go to feed the people. Yeah. And the second question. Male and female, both of them feed blood, like I don't have background about this kind of bug.
The people also be infected by other mechanisms. For example, I know that in Brazil, when the people drink juice in a rural condition, okay? Then not only the disease could be acquired, but this sucking blood insect, okay? The mother infected could transmit this disease to the child also, okay?
I also have a couple of questions. Do you know what type of T. cruzi is found in these bugs? Because there are several, type 1, type 2, type 3. Yes.
Yes.
There are all 7.
There are many strains of the glucid. The glucid that reproduced in Mepraya is asimodema different that this parasite that you collect in the Teratoma infectus. They are very different strains. By electrophoresis, you can separate these different strains.
But in Triatoma, you can find different isolates. Like actually 7. There are 7 big groups of T. cruzi, and you're saying that you can find all 7 of them in.
I think that is very important. There is not only molecular study in the I think that is important, realized not only molecular study in the parasites, in Trypanosoma cruzi. It's important, realized that realized Chagas with Trypanosoma cruzi. It's necessary to study the morphology, also the morphology of the parasite. Okay? In evolutionary perspective, it's very important the molecular study, but more important, I think, is the morphology of the species. Okay, the species are described for morphology, not only for DNA. The DNA is very good tools in evolutionary perspective, but morphology is more important to describe new species. Then, in relation with your question, I think that in this moment in Latin America, it's very important to study the morphology of the parasite. Okay, probably there are many species of parasite of Trypanosoma cruzi, or not only one species in all Latin America. Probably many species. There are about more to 100 species of vector, 140 species of vector, but only one species of vector of Trypanosoma cruzi. I think that also many species of the cruzi if we study the morphology of the cruzi, not only different.
Oh, that's an interesting point of view, and at some point people discussed it. So obviously T. cruzi is so polymorphic, so different. Type 1 is so different from type 6 that they should be considered as different species, that for sure, or even different genera, because they are so different and polymorphic. But consortium was assembled at some point, and they came to conclusions that because they cause the same Chagas disease, so let's call them T. cruzi and be done with it. Let's not confuse the world around us. That's why they still call the same species.
The problem is that the T. cruzi have a very complicated vital cycle, very complicated.
Yeah.
Thank you very much for.
Oh, I'm sorry. I have one more question. Sorry about that. But again, a more general one. If you compare the percentage of insects infected in your study, so you studied Mepraya. If you compare it to Triatoma, so where the percentage is higher, in Triatoma or in Mepraya?
In Mepraya, I think.
So there are more infected specimens.
No, well, infestant is the more important vector in Latin America and also in Chile. Okay, the difference is that in Mepraya, they when food, it'll go rapidly of the host. In infestant, not. This is better vector. Infectant that Mepraya. Okay?
Thank you very much. If there are no more questions, again, thank you. Thank you, very interesting talk.
Thank you.
And Our last speaker of this morning half before the coffee break is Dr. Yahir Otifai from Egypt. And he will talk about MCR, which is a movable computer ruler, as a new method for measuring the dimensions of parasite particles and other objects using optical microscope and the digital images.
It's okay. It's okay. لا تمام. It's okay. Hello all. I am Yahya Otifi, Professor of Parasitology. I have we can take a brief about the other methods. First, ocular micrometers, second, screw micrometer eyepiece, third, electron micrometer eyepiece, and image analysis systems. These methods is before the new methods we can reach it. This method applied while using using optical microscope. We can take some brief about the traditional methods for measuring. The instrument of ocular micrometer, micrometer inserted in the tube of eyepiece, and it contain eyepiece line, and it need the micrometer slides for adjustment. The second method, screw micrometer eyepiece. It inserted, remove the eyepiece tube of eyepiece and they put this screw micrometer. Both ocular micrometer eyepiece and screw micrometer eyepiece contain eyepiece lines. These eyepiece lines first must be adjusted with a stage micrometer or calibration scale. The adjustment depend on objective length 4, 10, or depend on objective length. These techniques of this method take more effort and stress on eye and consume more times and energy of the researcher. The new methods, movable computer ruler, MCR, applied on digital image and using Microsoft PowerPoint and Microsoft Picture Manager program. The materials of these methods, optical microscope, a digital fixed camera attached with fixed camera, optical microscope and fixed camera attached with PC or laptop, and programs, Microsoft programs, PowerPoint, and the slide parasites or any particles we can need to measure it. Beside the stage micrometer, our calibration slide. The steps, first image processing of parasite, image processing of stage micrometers, and design movable computer ruler, application of these methods, and measurement of parasite using other methods to compare it. First, photography and image processing of parasite. Taking the image of selected parasites with different magnifications. For example, toxoplasma taken by X10 and 40 and different magnification. Toxoplasma taxoid fresh samples, the image by 100 objective lens. Other types of parasites, eggs, fresh eggs of gastrointestinal nematodes, taken by 40x. Also, we can take the image of fresh parasites, fresh larvae of nematodes. Also, we can take the nematode larvae fresh by other optical magnifications. Other types, larvae of nematode, fixed and fresh larvae and eggs. Second step, photography and image processing of stage micrometer or calibration scales. There are different types of calibration scales, of calibration glasses. All these glasses contain scales, 1 millimeter, and divided to 10 large unit, and one of them subdivided to 10 sub-units, and each unit about 10 micro. We take the image of these scales by different objective lens. The image of parasite and the calibration scales must be passed in fixed condition. The fixed condition first is use the same microscope and same objective lens, use the same digital camera, use same density, pixel width and height of image, and the image should not be processed in other programs before taking the measurement. The image of calibration scales inserted in PowerPoint slide, and as we've seen, the large part is 100 micron, and small units 10 microns. Second step is making, how can we making the MCR, or movable computer ruler, the image of calibration scales cut by, cropped by, in the toolbar tools, crop the image and leave 1/7 of the image. This part we called rural. This part we insert round dot 3bt and the movable pointer. Insert lines, we make it as a movable pointer, and we must insert round dot 3bt. And how can we insert this dot by the office, PowerPoint office? This step is the movable computer ruler. How can we use it? And first we determine the size of dot, every dot. We determine the size of every dot by divided the 100 micron over the number of dot in this area. The number of dot is 338, and the result, one round dot equal 2.63 micrometer. Also, we can make different micrometer ruler according to the objective lens used. Application of MCR method. The image of parasites, this is a toxoplasma cyst image taken by 40 objective lens. And this MCR taken also by X40, and we can superimpositions of each image, and we can calculate the distance here, and the result is 32.63 micron. And we can repeat this image with different magnification, X10, X40, X100. The result of X100 is more accurate, and because we show more details of the image. Also, we can use it in small parasites like Toxoplasma tachyzoids. First, we determine the parasite, we can measure it, and we can show the length and width. The length and width of this Toxoplasma tachyzoid is 7.5 multiplied to width 2.5 millimicron. Also, we can use it in small details in oocyst, Eimeria oocyst. We can measure the steady body sporocyst and length of wall and total length. More details, all details in oocyst, we can measure it by this method. Also, we can make two MCR in the same slide, and can use it in more MCR in the same slide. We can make it. Trichostrongylas eggs by X40, length about this 98.63 micron. Also, we can create scale bar. By formation of MCRs, scale bar is easily created and attached to the image of parasites. This is the MCR at 100. A line space is 10 micron, and we can make scale bar. Scale bar, this scale bar is 10 micron. Also, we can make a scale bar by different magnification by using MCR at different magnification. Determine this line and insert it in the pictures. Scale bar also. We can make, we can create a graduated scale bar. Protostrongylus species from the sheep, first larva. This image by X40 and the MCR by 40. We can superimpose it, and each unit here is 10 microns. This is a graduate scale bar. This graduate scale bar and the image fixed on the image. Also, we can make graduated scale bars on different eggs of parasitic gastroenteritis. Scale bar also of different type of oocyst, sporulated oocyst of goat. This is scale bar, graduate scale bar. A research summary. MCR is a new method for measuring the size of parasite and other objective added to other method. MCR is designed, its design is derived from digital image of a stage micrometer. It can be superimposed on image of parasite to measure their size, height, pixel number, and density are fixed for every image. MCR method saves time and effort. Conclusion, MCR have been tested using selected parasite. It has been shown to be as accurate as the other method. The image must be from well-prepared specimen, good focus and good quality. The image should not be edited in other program period taking the measurement. This method is less complex and more precise. Applicable even in laboratory with limited resource. Save effort and cost. Easy and fast in application, can be used every time you need. MCR is efficient techniques. The scale bar also will be easily created and attached to the image. For more details who are interested, experimental parasitology, these papers, more details of this methods. This is an image of picture of my faculty and my lab. Image of my city, Alexandria University, Egypt. Thank you all very much.
Thank you very much.
Thank you.
Questions? Yes. I got it.
Thank you very much for your interesting work. One question. For your scale, it's easy to measure the egg or some cyst and so on. But for the curved navel, because it is very like this, how to measure the length of the navel?
We can move the scales. This MCR we can move in any area in the image. We can take the image, take the diameter. We can move it. There is a slide here. We can.
Yeah, this one.
I can move it. Please, I can help. I need help to move this slide. Help. Small size.
You want to make it small?
Yes, I can move. Can I move this? Also, can move this slide to any area we can need to measure it.
Okay, I see.
Yes.
Thank you very much.
Yes, thanks.
Hello. I'd like to appreciate your great presentation. It's very instructive and very simple. And I also do appreciate that you put the photo of Alexandria University and the photo of the city. I do have a question. This MCR method, is it a well-known method? Sorry, I missed a little part of your presentation. Is this a well-known method and you modified it, or you created from the beginning?
The new method.
It's a new method.
New method. I search in the reference, different reference. I cannot reach to any details about it. This is a new method.
Oh, that's
for
a great work,
measurement.
actually. It's a great work. I do
Yes.
appreciate your work.
Yes.
Thank you.
Thank you, thank you.
More questions? Thank you very much.
Thank you, thank you,
Thank you.
thank you.
Before the coffee break, I have a few more announcements, and actually one pleasant thing to do. I need to distribute diplomas for keynote speakers of the conference, and I would like to do all at once. So there is a certificate of recognition that goes to Dr. Shen for the phenomenal and worthy keynote presentation at the International Conference on Parasitology.
Is the candle it or you need it or I can delete it?
I can delete it. Is this it?
You-
I can delete it. Delete it.
Dr. Lasser, that's a
Is that you?
diploma of recognition for the phenomenal and worthy keynote presentation. Thank you.
And deal this PowerPoint.
Dr. Otify,
Yes, thanks.
that's a diploma, certificate of recognition for the phenomenal and worthy presentation.
Thank you.
Thank you. Yep. And well, we're not done yet. We have the whole part right now. Those diplomas go to the members of the organizing committee, scientific organizing committee of the conference. The all will be in appreciation of real support as a member of the organizing committee, because without those people, the conference would not happen. End of the story. The first diploma goes to Dr. Carriero. Dr. Hyde. Thank you. Dr. Papayeva. Dr. Shen. Thank you. Dr. Xu. Can you help me with reading the name correctly?
Dr. Xu Jiannong. I don't know him.
He's not here yet, so we'll leave it. Well, that's me. And to Dr. Hutter. Thank you very much. Now, guys, we have the last announcement, and you will have your coffee break. The last one is that there will be a group photo at the end of the next session before lunch break. So we will all gather together here to be photo taken, and then after that, there will be lunch. Now, we are very, very good in our schedule, so I suggest we take, well, we can take 15, we can take 20 minutes break. We can take 20 minutes break, and we can start our next session at 10:55. And hold on one sec, there is one more announcement.
Okay. I'm Dr. Harim Khater. I've been at a conference of WAF, that's the World Organization of Advancement of Veterinary Parasitology in Liverpool last week. And I met a lot of people there, and we decided to make a subgroup of the organization for the Arab and the developing world. We call it Arab World Association of Development of Parasitology. It's appreciated as AWAB. So we made a meeting at that conference actually, and we collected the names of interested people to share their ideas. Our main goal is to to discover, there is a language barrier for the Arab speaker and also scientists who are not native English speakers. So their work, they usually publish their work in a local journals, which nobody knows about them. So our main goal is to explore their work to the world, or at least by submitting their abstracts to our websites or helping them to publish in more impact factor journals. And we will do also public orientation about parasitology. So this sub-association will get the fund from the main group, which is WAF, and we will get international fund actually. And this will be a great work for researchers who are not speaking English well, or they do have a language barrier for presenting work in highly cited journals. If you are interested, please give me your names and your contact numbers, and we will update you and to be a member of our new subgroup, sub-association from the WAF. Thank you, appreciate your listening. Thank you.
And well, in 20 minutes or whatever, we can actually meet at 11, just to be, that's a good number. Let's meet at 11. And the chairman of the next section is Dr. Hyde. And would you like to say something?
Hello, Professor Carniero and myself are chairing the next session. Could we ask the speakers for the next session, please, just to come up before the coffee break, so that we can just check your presentations are okay and that you're here. Thank you very much.
And actually, it's a good idea for all the speakers. It's a good idea to double check whether your presentations are uploaded, where everything is working correctly. So lunch break seems like a very good time to do that. Okay? So coffee break.
very much.
First of all, I want to thank the organizing committee for inviting me to this presentation. I will talk about an epidemiological work, the role of environment in the spreading of visceral leishmaniasis in the western São Paulo, Brazil. Visceral leishmaniasis, VL, is an emerging zoonosis distributed worldwide, though geographic distribution is mainly in tropical and temperate regions. However, 5 countries worldwide harbor greater than 90% of the cases. In South America, the vector is Lutzomania longipalpis. The parasite is Leishmania shaggsi, and dogs are the main reservoirs, and humans are us. All of them are involved in the biological cycle. Brazil harbor 90% of VL in Latin America. Nevertheless, most of the cases are from the Northeast region. However, in the last years, 5 regions and 21 of the 26 states of the country were identified infected dogs and humans. Recently, in 2015, the disease reached the South region, a free region of VL. 2 months ago, the infected dogs were identified in Foz do Iguaçu, in Paraná State, in the border of Paraguay. In São Paulo, a free area of VL, the first cases were described in 1999. Different from other regions of Brazil, such as Minas Gerais and Espírito Santo, the parasites came from Bolivia. There are several evidences showing this fact. Here we can see the world distribution. 5 countries harbor most of infected individuals. Brazil, North and South Sudan, Ethiopia, and India. Brazil is the biggest country of Latin America, has a continental dimension, and it was suggested that in São Paulo state, the disease came from Bolivia, not from Northeast. From Santa Cruz de la Sierra, in the border of Mato Grosso do Sul, and the disease came to São Paulo state. São Paulo state is located in southeast region of Brazil. But we live in the west region of São Paulo state, a free area of VL. The VL route, as I told you, it suggests that the disease came to São Paulo from Bolivia. Many facts are involved in these suggestions. In 1952, a railway construction from Santa Cruz de la Sierra to São Paulo state was effected. Many years after, a highway it was constructed near the railway. But recently, the Bolivia-Brazil pipeline gas construction was effected. And so we have a railway, a highway, and a gas pipeline, one near the other. Infected dogs and individuals came from the border of Brazil, in Mato Grosso, from the capital of Mato Grosso, and they arrived in the border of São Paulo. The Western region was a free area of VL, but in 2005, the first case was described. Several authors showed the infection in São Paulo state, from Bolivia to Mato Grosso to São Paulo. The gas pipeline, the railway, and the highway. In these counties, there are infected dogs and infected humans. But there is no description about the infection in the West region. Our objective was to describe the role of environment in the fast worrying spread of VL in western São Paulo state. We hypothesized that the primary axis of VL dissemination through the western region was the SP 560 highway. This highway, coming from endemic areas of Mato Grosso state and throughout the whole region, crowded by small and middle cities and connected by a large number of highways. The healthcare regional 11 comprises 5 subregions and 45 counties. In one of the subregions is Pontal of Paranapanema, the poorest region of São Paulo state. VL affects the poor among the poorest. And so, besides poor people, we have some environment risk factors. For example, we have a sandy and dry soil in the winter and a rainy and wet soil in the summer. There are increasing temperatures in last decades with an average of 23.5 degrees. Nevertheless, the main characteristic of the region is the extensive amount of watersheds flowing into Paraná, Paranapanema, and Tietê rivers. These are the 3 biggest rivers of southeast and southern Brazil. These rivers harbor 9 artificial big lakes, and the flood area of 2,384 square miles. These big artificial lakes support 9 hydroelectric plants and bridges, linking endemic regions to non-endemic regions. Here we can see the main slide of the presentation, because this is the infected area, the gas line, the railway, and the highway. These counties is crowded of infected dogs and individuals. But the western region, there was not infected VL dogs. We suppose that the disease spread in the west region through this highway, because this highway comes from endemic regions to free areas. This region is crowded of small counties. These counties are linked to small highways supporting middle and little cities. Besides, we have the big artificial lakes, the Tietê River, the Paranapanema River in the border of Paraná State, and in Paraná River, the biggest river of southwest Brazil. These lakes, these artificial lakes, support 9 hydroelectric plants for electrolyte generation. In these lakes, there are several bridges. By these bridges, humans, beings, and infected dogs goes from endemic to non-endemic regions. The route of sandflies. In Brazil, the vector is Lutzomania longipalpis. We suppose that in West region, the transmission occurs by contiguity. In our work, the presence of vectors in infected dogs and humans were determined in 3 years interval. Here, the spreading of the disease. First, the sandflies. In this period, 2002 to 2004, the disease, the infected sandflies were present here in the border of the highway. In the next period, 2005 to 2007, by contiguity, the sandflies, infected sandflies, reached these counties. In the border, this is the secondary axis, a little highway. In this period, the disease spread from this area to far from here, in the border of Paraná state. And by contiguity, following the secondary axis, the infected sandflies were found here. Finally, the explosion of the disease happened in the last 3 years. In this time, a lot of counties were infected sandflies were found by contiguity, following the primary axis and the secondary axis. Now, the canine VL footprints. We suggest that transmission occurs by contiguity to the counties, and the presence was determined in 3 years interval. It's known that infected dogs and humans appears 3 to 5 years after sandflies. I showed you before that sandflies, they were here in the next slide, but no infected dogs exist here. But now, 4 years after, the infected dogs followed the same standard of the sandflies, and the region is crowded of infected dogs, following the primary axis and the secondary axis by contiguity. In 2008 to 2010, so far from the beginning, we see infected dogs in the border of Paraná, following the primary axis and the secondary axis. And finally, in 2011 to 2013, by contiguity, infected dogs were found in secondary axis and the counties by contiguity. After sandflies, dogs, the humans appeared. As suggested before, the transmission occurs by contiguity, and the presence was determined in 3 years interval. Following the same standard, we don't have infected dogs and humans, but 5 years after, we see the same standard of sandflies and dogs. And there are several countries showing infected individuals. In the primary and the secondary axis. In the next period, 2008-2010, the disease spread, and in the last 3 years, all these countries showed infected individuals in the primary axis and in the secondary axis. Now, we can say that the western region of São Paulo state is a brand new endemic area for VL sandflies. Why? Because in 12 years interval, 66.7% of the counties of the western region showed infected sandflies. The same situation, it's about infected dogs. In 12 years interval, 40% of the counties of RRAS 11 showed infected dogs. And finally, after sandflies, infected dogs, the humans. In 12 years interval, 31.1% of counties of RAS human VL. In this period, 348 cases and 19 deaths were related. Conclusion, we can say that poverty, climate, extensive highway network, artificial big lakes linking endemic regions may endorse environment factors related to the spreading of VL in western São Paulo state. This is the VL team of Presidente Prudente. We have some collaborators, Dr. Dilton Moraes from Universidade de São Paulo. Elivelton Fonseca is a postdoctoral student from Louisiana University. And Ivete da Rocha, she is the head of SUSEM in São Paulo state. Here, the regional hospital of Presidente Prudente that supports infected children. All the children of RAS for this region are sent to this hospital. It is a public tertiary university hospital with 450 beds. The West Paulista University is the biggest university of West region of São Paulo state. Thank you very much.
Thank you very much for a very interesting and clear presentation. Are there any questions?
My question is, there are other wild mammals with this parasite?
Yes, I didn't talk about this, but in fact dogs are the main, but there are wild animals. In the region, there was not research about this, but in the state, in Brazil, there is. Okay.
This could explain the migration, this disease in São Paulo, maybe, no?
There are several evidences that the infected dogs came from Bolivia and not from Northeast region. For example, in Minas Gerais, Espírito Santo, it's known that the infected dogs and infected individuals came from the Northeast region. However, in São Paulo state, it's very different. And now, recently, 2 months ago, it was identified a focus on Foz do Iguaçu, in the south region, a free area of VL. They don't know why and where the infected dogs came from Foz do Iguaçu. If they are autochthonous dogs, but yes, we don't know from where the parasite came in Foz do Iguaçu. In São Paulo, probably from Bolivia.
And the last question, which is the origin of this parasite? The origin in Latin America? Is in Bolivia?
From São Paulo, yes. Yes, in Bolivia there are some places with visceral leishmaniasis. In Paraguay also.
Thank you very much.
Okay, we do have a busy morning this morning, so perhaps we could make this one the last question, please. Thank you.
How do you know they're from Bolivia and not from the neighboring states of Brazil? Why?
There's a very interesting paper published in PLOS ONE some 4 years ago that they proved the root of infected individuals working in the railway, in the highway, and recently in the pipeline gas construction. And they showed exactly how the disease walked from Bolivia to the border of Brazil in Corumbá, after the disease came to the capital, Campo Grande, and after they showed the presence of the infected individuals in the border of São Paulo. They stopped here. Some researchers start in São Paulo and showed how the disease is getting to the capital. There is a lot of papers published, but no papers in the West region.
Thank you.
Okay, thank you very much. I'll have to call time on the questions there. Perhaps you can have a chat with the speakers at lunchtime. Okay, our next speaker is Dr. Sonia Almeria from the University Autónoma de Barcelona in Spain.
Where do I find it? This is the point. Sonia. Okay, thank you very much. Good morning. Today I will be talking about a veterinary parasitology parasite. I'm talking about bovine neosporosis, and I will do a little review of epidemiology and the markers of risk of abortion from a clinical perspective. Neosporosis is caused by Neospora caninum. It's a parasite very close related to Toxoplasma gondii. Was first discovered in dogs in 1984, and a few years later was discovered causing outbreaks of abortion in cattle in 1989 in the USA. However, it's not considered a new disease. It's a problem of diagnosis. It was believed that was not diagnosed properly before. Since it was described, the disease has been mainly observed in ruminants, in horses, and also in dogs, but it's mainly a disease of cattle and dogs. And in cattle, that is what I'm talking today, is considered today, like nowadays, one of the main causes of abortion worldwide. In the USA only, like the cause of this infection abortion is considered around 500 million per year in a recent study, only in dairy cattle, but it's also affecting beef cattle, so it's even more a problem than this. The parasite is very close related to Toxoplasma gondii, and as you can see, like this is Dr. Dubin, that is the discovery of the parasite, and if we change in Toxoplasma gondii the definitive host, the cat, by the dog, the sign of the roots is very similar in the parasites and the tissues and all the stages, but it's not a zoonosis. Neospora doesn't affect persons, And while Toxoplasma causes abortion also in humans, but also in animals like sheep, but not in cattle, Neospora is the cause of abortion in cattle. So the difference is in the host mainly. In cattle, the Neospora's abortion is also has certain characteristics. It's an abortion that can occur in one animal once, but importantly can be repeated, which doesn't happen in Toxoplasma. We don't have protective immunity against repeated abortion in Neospora. And the animals affected are animals of any age, but it's most common around the 5 or 6 months of pregnancy, in the second trimester of pregnancy in the animals. On top of abortion, we have the appearance of mummification sometimes, birth of calves that die very few days afterwards. We have also paralysis in weak calves, like this image in the limbs. And we don't have a treatment, there is no adequate treatment, and there is a lot of efforts to create a vaccine, but there is no vaccine available nowadays. So our efforts are done to control the disease based on herd management. And to do that, we need to know which animals are infected. The way to know if a cattle herd or which animals particularly are infected is through ELISA, the detection of antibodies. And then we have to check the abortion to check that the parasite is in the fetus. And to check the herds, we have available many ELISA kits that are all very good. And in the very recent study was shown that all of them had high sensitivity or specificity, so it's our tool to work on them. Based on our studies, I'm going to summarize main epidemiological findings that are in agreement with other authors. The first importance, and very different to Toxoplasma, is that Neospora antibodies are related to abortion. So a seropositive animal is not protected, is an animal that can abort and has high risk of abortion. If in a herd you have this percentage of abortions, in seronegative animals, normally there has to be around 4 or 5% maximum. In seropositive animal can reach 20–30% easily in infected herds. And the higher the titers, more abortion risk in these animals. So contrary to a protective immunity in Toxoplasma, Neospora doesn't have a protective immunity when one animal is infected, and on the contrary, this animal has higher risk of abortion. Similar to Toxoplasma, we have a very high rate of vertical transmission. It's up to 95% in the infected animals, and although some animals will abort, most of the animals will have a descendant that will be seropositive, and if it's a heifer, will have possibilities of abortion. So the high rate of abortion is very important in the herd to maintain the disease and to cause trouble. We have also studied differences between beef and dairy cattle, and it seems that in the beef, during gestation, the antibody levels are always higher compared with crossbreed animals that are beef and dairy mixed, and then dairy animals have the lowest titers of these studies. So it seems to be some differences there. In our studies, importantly, we have seen more or less that around 40 or 30% of the animals until 40 of the seropositive animals abort. So we have seropositive animals, we know that a certain percentage of those animals are going to abort. And this is no difference in parious cows or in heifers. It's independent of the lactation number of these animals. Also, we have seen that it's quite constant, the animals or percentage of animals that abort, because if we have different herds like these ones, the percentage of prevalence was from 26, 28, but one only had a 4%. However, the percentage of animals that abort that are seropositives is always around 25–30% of the animals. So it seems that there is always that percentage of animals that will abort. The timing on abortion, if an animal is chronically infected before in a herd, because we have a certain percentage of seropositive animals, we have seen that the abortion will never take abort before 90 days of gestation. There are other factors like season of parity that will be important then. But after 90 days of pregnancy, if the animal is seropositive, that becomes a probability of abortion of an odds ratio of more than 12% compared with seronegative animals. So after 90 days, if an animal is seropositive, the probability of abortion is high. good for the farmers. We don't have any affection of the pregnancy rate, and fertility is not affected. So in seropositive and seronegative animals, after an abortion, the animal recover and can get pregnant without any problem, and the fertility is not affected. So that's a good point. And another important point of our studies has been that to try to control the seroprevalence in the herd, we told the farmers not to keep seropositive animals. That is the only way not to maintain in the next generation the parasite. And to do that, we told the farmers to inseminate with beef cattle, so the reposition of the herd wouldn't stay in. And our idea was to try to reduce the seroprevalence, but also what we found is that actually the percentage of abortion in these crossbred pregnancies, particularly if we use the semen of a Limousin herd bred that is a French bred, we got that the percentage of abortions was very low compared with seropositive in the Frisian pure animals. So now we are using this control measure to control the prevalence and reduce the abortions in the herds. So as main conclusions of these epidemiological aspects, I will say Neospora doesn't cause abortion after 90 days only abort after 90 days of pregnancy, does not affect the fertility of the animals, seems to be higher susceptibility in dairy than in beef breeds, but the use of semen from the beef breeds not only reduce the number of infected herds that will not be used as replacement, but also reduce the risk of abortion, especially when using the Limousin semen. And this is now considered one of the control measures worldwide to be used in this parasite. About what caused the abortion is not really well known, but of course the parasite has to multiply in the placenta or in the fetus and cause enough damage so the fetus is not viable. But it has been shown that the maternal immune response is very important also in this abortion, and that an excessive and Th1 immune response will actually be also causing part of this abortion. So the maternal immune response also is affecting the abortion. What we try to find now is, if we know that a certain percentage of animals will abort, we want to try to know which animals are going to abort to try to reduce these animals in the herd before cause more economical damage. And to do that, what we have analyzed is first the role of interferon gamma with the analysis in the plasma, and we try to check the interferon gamma production. And this interferon gamma seems to be a good marker of abortion, since the presence of positive production in seropositive animals only reduce the presence of abortion compared with animals that were seropositive and not producing interferon gamma that had a very higher rate of abortion, a 6.3 versus the 29% usual found in these animals. So we can say that the interferon gamma and up to a threshold is a positive influence in this disease. Another marker that we have found useful is what we call the pregnancy-associated glycoproteins, PAGs. There are two types, PAG1 and PAG2. And we analyze this because PAG is considered a marker of placenta well-being in animals. So when the animal gets pregnant, these proteins start growing, appearing in the pregnancy, and they increase through pregnancy. We observed that in the non-aborting seropositive animals, there was no differences in the levels compared with seronegative animals, and the increase in the normal rate. So it seems that if the animal is not aborting, the placenta is not damaged, and there is not enough abortion. So at least we know that the PAGs are still increasing, the pregnancy is coming through. While in animals that abort, we can see that the levels start, these are 3 animals. This one just maintain the levels, but these ones that have aborted fetus, clearly the PAG levels start decreasing and became 0 because the placental damage was so big that now abortion occurs. So we think that the PAG can be a good marker also to know which animal will abort and which one won't. Interestingly, very interesting for us is that we observe also that the PAG levels are always higher during pregnancy in animals that are crossbred. So those animals that we told the farmer to inseminate with beef semen have higher levels of PAG levels compared with the Frisian animals. And we don't know why, but this seems to indicate that the placental bean in these animals, in the crossbred pregnancies, is more resistant to the parasite, so there is no abortions in these animals. The other marker is a PAG2. PAG2 is another pregnancy-associated glycoprotein. This one, we have done a recent study, and we observed that the animals that have like at 120 days of pregnancy, we chose that time because it's before when the abortion usually takes place, and we observed that a certain level is indication or not of abortion, because if we have minus 4.5 nanograms per milliliter, these animals are going to have a higher risk of abortion compared with animals that have higher protein PAG2. So we are trying to find out if this is, we have to confirm with more studies, if this PAG2 is really like a good marker to know which animals at 120 will abort or not, and tell the farmer to go ahead with pregnancies or lactations or what need to be done in the herd. So as conclusions of this second part, I have said that the Neospora antibody titers are indicator of not only of seropositivity but also of risk of abortion, and we need to do a titration. Not only like consider animals seropositive, seronegative. We need to know which is the titers, because the higher the titer, the higher the risk of abortion. That interferon gamma and crossbred pregnancies can protect Neospora infected dairy cows against these abortions. And although Neospora infection seems not to affect the placental function in the non-aborting cows, we can try to use this PAG2 concentration to predict abortion at 120 days of pregnancy, to see if that way we can control the disease in the herds. So, and that's all. I would like to acknowledge the very excellent collaborators that we have had in these studies, and some of the Spanish grants that have supported our study. And thank you for your attention.
Thank you for a very interesting talk. Has anybody got any questions?
Thank you for your nice presentation. And the neosporosis is also reported from time to time in China.
Mm-hmm.
Now, based on my knowledge, it's just second to the rotavirus in children, diarrhea.
Mm-hmm.
So it's very interesting. My question is why the antibody titer is higher in the beef cattle than in dairy cattle? Is there any, what causes these differentiation?
We don't know. We analyze the titers in the same herd, in the same management, and we don't know why they're higher, because cattle usually doesn't report as much as dairy cattle, and in dairy cattle is clearly a titration level, that the higher the titers, the more abortion, but that doesn't seem to be true for beef cattle. We don't know the reason.
Okay, thanks.
Thank you.
A question at the back there.
Hi, Sonia. Thank you for your presentation. I would like to know if these PAG molecules released by the parasite are related with the loading of the parasite inside of the host, or is it for virulence of the parasite? Thank you.
We don't know that. We have done now an analysis in experimental infected animals to check if there is any relationship with the parasite load, but it's not really known, because we don't really, it's not easy to know how many parasites the cattle had. We only know the titers of the antibodies, but we cannot relate exactly which is the level of the parasite. We suppose that the more replicate of the parasite in the animal will be higher titers. If the PAGs are related to the parasite levels, we believe that will be, but we don't know why at 120 is such a limit, a time to know which way the pregnancy will go. We need more studies to know that.
Okay, thank you very much. I think we moved on to our next speaker now. next speaker is Dr. Stefan Curvers from the Zuiderland Medical Center in the Netherlands.
Do you have a pointer? Oh, here it is.
So that's the light, and that's forward and backwards.
Oh, okay, that should be fine. I want to thank the committee for inviting me to have this presentation. It's about a case report that we experienced in the lab I work in 2012, so it's a little years back. Strongyloides is a silent killer. You all know the parasite. And I would start with the patient is a male patient, 57 years of age, and a region from Liberia in Africa. I want to first start with a timeline as a medical review, and we have 4 different episodes. We'll start with medical history, May 2011, April 2012, and May in that same year. As I said, the patient was originally from Liberia and was living for 9 years already in the Netherlands when we saw him in 2012. I'll start with the medical history, and he was known with chronic hepatitis B and a diabetes 2 condition. And the episode in 2011, he has a diagnosis of a non-Hodgkin lymphoma, which was treated with chemotherapy at that time. When we saw a month earlier, 2012 in April, he was hospitalized with abdominal and respiratory complaints with symptoms like vomiting, nausea, and fever. And that fever remained despite of the broad-spectrum antibiotics which he was treated with at that time. Also, there was an infiltrate shown at the thoracic X-ray, and at that time there also are some lab results done, which we see that all have some lower than normal, like the hemoglobin, hematocrit, red blood cell count, red blood cell count, like neutros, monos, limbs. EUs are at a normal level, and the CRP has raised. And at that time, the doctors think of a pneumocystis pneumonia. There was a suspicion of it, and they treated it with. The patient was discharged from the hospital in a good clinical condition. But despite of that, in 2012, a month later, he was again hospitalized due to general weakness and also abdominal complaints. And the symptoms now are headache, diarrhea, vomiting, nausea, and fever. So it was not going that well with the patient. But after extensive diagnostics examination, no cause was found, however. But after 14 days, because of that persistent vomiting, what still was occurring, a gastroscopy was performed, and the following was seen. It was an ulcerative lesion. The patient had 2. So it was shown here at the red border of it. The picture isn't that clear, but I had to make it bigger in size. I hope it's clear for you to see. And during that gastroscopy, biopsies were taken, and after preparation staining at the pathology department, we saw this. was a hematoxylin eosine staining at a magnification of 1000. And when we enlarge that, it looks like this. And we have a closer look. There were worm larvae-like structures seen. And at this point, and because of these findings, our department was consulted for what is this? Of course, there was a suspicion, but before that, a parasitological diagnosis was made. The patient deteriorated and was admitted to the ICU, where they started medical ventilation and broad-spectrum antibiotics. Also, collecting stool at that time was very difficult because the patient was suffering from an ileus. So that was a problem, but eventually stool samples were collected, examined for parasites, and this is what we saw at that time. They're all wet mounds. And the measurements are 250 micron in length and 17 in width. Of course, there was, I hope it's good to see in the back, a genital primordium. And at last there was a short buccal cavity. So as a conclusion, we can say these are of a Strongylodus stercoralis. Now we know this, the ivermectin therapy was started right away, but one day after the diagnosis was confirmed, the patient died of a septicemia with a massive bleeding from the stomach ulcers, so the diagnosis came too late for this patient, and the conclusion was a disseminated strongyloidiasis. Now we can have a discussion for this patient and could this be prevented? Maybe not, but there was no awareness of strongyloidiasis due to the lack of eosinophilia, what was shown earlier in the lab results. And the patient was living for 9 years in the Netherlands. There was no recent visit to an endemic country. Therefore, there was never serology performed before the immunosuppressive therapy was started. So perhaps had that been done at the time, maybe they were aware of the strongylosis infection, what was going on at that time. Also, there was never a collection of a bronchial alveolar lavage was not done. Why? That is the question. Perhaps the doctors at that time don't think it was necessary. And there are the guidelines for screening of strongylosis concerning patients who will have immunosuppressive therapy. In our country, there are none. So I don't know how that is globally situated. Perhaps that is a question to you. Some short facts on strongylosis, the life cycle. Of course, you see the filiform larvae penetrate the skin and go straight away to the lumen where they grow adult. And then eggs which immediately hatch, and then the larvae or go back into the natural cycle out of the body, or they cause an auto-infection. And when the patient is immunosuppressive, that will go very fast. Therefore, the transmission occurs mainly in tropical and subtropical regions. There are 30 to 100 million people infected worldwide, but precise data on the prevalence are known in endemic countries. And strongylosis is frequently underdiagnosed because many cases are asymptomatic, or moreover, the diagnostic methods lack sensitivity. Without the appropriate therapy, the infection does not resolve and may persist for life. And infection may be severe and even life-threatening in cases of immunodeficiency. No global health strategies for controlling disease are active at that time. I want to say some take-home messages. Healthcare providers should be made aware of this parasite, and particularly about the risk of this disseminated infections, because you see in the study I did before this, I read a lot of literature, and cases like this occur all over the world, and a more frequent. Perhaps there should be a standardization of screening for strong gonorias before starting immunosuppressive therapy using serology or even PCR concerning high-risk patients. Perhaps that will lower down cases like this. And a more detailed epidemiological data on the global distribution of strong gonorias are needed. I want a special thank to the microbiologists, which are my superiors at the lab, and of course the other doctors which were involved in treating this patient at the time in 2012. I want to thank you for the attention. And are there questions?
Thank you very much for a very clear and interesting presentation. Any questions, please?
Stefan, thank you very much for a stimulating delivery. My question is this. You found rhabditiform larva in the stomach. Are we sure it is rhabditiform and not filariform? Because it is the filariform one that is actually invasive, and it goes in through the skin when it is transmitted to humans.
About the rhabditiform larva we found in the stool, I give you why we think it was a rhabditiform. Eventually, there was no evidence for finding filariform because there was no ever sputum or respiratory material. Where do you think that the filariform larvae should occur in? So that is unfortunately not done. So I was hoping to get that, but the patient was already dead.
Thank you very much for the nice presentation. My name is Dr. Cohn from New Jersey. I have a few questions concerning the case. I was wondering why you guys suspected PCP. I didn't see anything pointing towards PCP. I don't know whether you guys went ahead to do a bronchoscopy. Was there an LDH? Did you do a CAT scan? Was there an ABG? What made you suspect PCP?
That is a good question, but I was not involved in it because I am the lab technician who do search for the parasites in the stools that we get. But what I know is that offspite the patient was not responding to the broad spectrum diet, broad spectrum antibiotics. The fever remains, and therefore the infiltrate was shown on the X-ray. They had a suspicion of PCP. There was at that time no ball done to see if it was actually a PCP, and they just started right away with the therapy of it. So I think the doctors made a mistake there, but I can't, and that a gastroscopy was done after that. The vomiting was taking place for 14 days, and I don't know how, I don't know why the doctors waited that long, but in my point of view, that is too long. But I can't give you any answer to that, why the doctors did what they did at this time.
Okay, I think we need to move on now, so perhaps we can have a chat at lunchtime.
Okay, thank you very much.
Thank you very much. Thank you.
I would like to introduce to the new speaker, student Dr. Geoff Hyde from the University of Salford, United Kingdom.
Okay, good morning. I want to talk about toxoplasma in my presentation this morning, and I'm interested in this parasite because of its ubiquity and because of how common it is in populations. It's probably one of the most prevalent parasites globally. About 1 in 3 people on average are infected globally, so in this room 1 in 3 of us potentially are infected. It affects a wide range of animals, all warm-blooded mammals, humans, domestic animals, and even marine mammals. And so it's a very, very successful parasite. And my interest really is thinking about why this is. It's also a parasite of the cat, and that makes it even more intriguing as to how the parasite can be so widespread despite having only really one definitive host. So my talk's really about exploring the possibility of vertical transmission from parent to offspring as a mechanism for promoting toxoplasma transmission. So I'm going to talk a little bit about the background to the disease, just what's relevant for the talk, and we've done some studies in sheep, rodents and humans, which I'd like to sort of talk about. So I've talked really already about these high prevalences of infection. The parasite is being a parasite of the cat, and it's very widely geographically distributed. Virtually every continent on the planet is known to have infections. Of course, from a human perspective, it causes serious disease, and therefore it's of importance. The major issues are causing miscarriage in humans, causing abortion in domestic animals, for example. It causes things like ocular disease, but in general, the infection is asymptomatic. So those 30% of the global population infected, the majority have an asymptomatic infection. This gives you an idea. Professor Shen showed this diagram already, so you can see the distribution of the parasite. In some countries, China and the UK, it's around about 10%. In the US and Canada, 10 to 20%. In some parts of South America and Europe, greater than 40% prevalence, so hugely important. And of course, an at-risk group is the pregnant women. This is a study that Professor Shen and colleagues did in China, looking at the distribution locally. And what you see generally, if you look across the globe and locally like this, you see differences in the prevalence, okay, which I find interesting. It causes abortion in sheep is one example of the effects of it. This is a friend of mine. It causes retinochoroiditis, degeneration of the retina, and you can see how it's a progressive disease over ages. But I should stress that primarily the infection is asymptomatic. A quick run through the life cycle to indicate the mechanisms of transmission. So the cat's the definitive host. It distributes oocysts into the environment, actually very many millions of oocysts, so it's very highly infectious when it is infective. It infects secondary hosts, which can be, as I say, all warm-blooded animals, including humans and even marsupials and marine mammals. A secondary route of transmission is by carnivory, so ingestion of raw meat from infected animals. And then finally, this third route of transmission, congenital transmission. So effectively, we've got 3 routes of transmission, and somehow those 3 routes of transmission have to explain the wide ubiquity of infection in animals and also these very high prevalences that we see in many species. So that's where my interest comes, is how important are each of these routes of transmission. Very difficult to measure in an environmental setting because we can't really measure many of these easily. So my research really is focusing on that third one there, the congenital transmission from mother to offspring. So we've got this dilemma. We've got the dilemma that the cat is the only definitive host, but the parasite is ubiquitous and found at high prevalences. So I'm interested in these cycles of transmission that effectively bypass the cat. And I want to try to gain some understanding of the importance of these. So I'm going to talk about our studies in three groups of animals, in rodents and in sheep and in humans, and try to, I hope, show you that perhaps congenital and vertical transmission may be much more important than is currently recognized for this particular parasite. So first of all, looking at rodents. So a number of studies I want to kind of cover here. So our first study, we looked at a population of mice. So I'm interested in naturally infected animals and humans. We looked at 200 mice that were trapped from an urban population. We used PCR to detect the parasite, and we were interested in analysing transmission in pregnant mice within this natural population. So this was a study carried out in Manchester, where I believe this conference is going to be next year. We collected these 200 mice, about 16 of them were pregnant, and we dissected out the fetuses, and we tested the fetuses for infection. 59% of the mice were infected with toxoplasma, which is very high. 16 of the pregnant mice were pregnant, and 12 of those were positive. And these were the results we got. And what we found was, when we looked at the 12 infected mothers, of the fetuses, there were 63 fetuses in those 12 mothers, and 74.6 of those fetuses were infected prior even to birth. Okay. Of course, the uninfected mothers produced no infected offspring, and when we looked at the population, as a whole, about 60% transmission was there. And what we found was that of all of those 12 infected mothers were passed on their infection to at least one of the fetuses. So we have this high prevalence of vertical transmission, and we would suggest that this means that the congenital transmission may be much more important than previously thought. We did a study on a captive population of mice. This was a population of mice that had been set up from a founding population, kept in captivity for a period of greater than 5 years, never exposed to cats during that time. And again, we tested some of those for toxoplasma. We looked at 89 mice, 76.4% were infected. So remember, this is a population of mice that have never been exposed to cats. We genotyped the mice, and of course they were highly inbred because of the nature of the population. And again, in this closed colony, we would propose that this transmission is caused by vertical transmission. In a third study, we looked at a wild population of wood mice. So these are species of Apodemus sylvaticus, the wood mouse. And again, we looked at a population that was collected from an area which was relatively free from cats. 2 of them were pregnant, but what we did was we tested these mice, 206 mice. About 40.78% were infected with toxoplasma, which is a very high percentage of infection given the relatively few cats in this area. 50% of the pregnant mice were infected. And again, we demonstrated a relatively high prevalence in these animals. So we did a comparison between the urban population that we studied initially, where we had 59%, and we knew the distribution of cats in the urban population by surveying the area, and there was something like a density of greater than 5 cats per kilometre squared, with 59% prevalence of infection. And in the area without cats, where we estimated it to be less than 2.5 cats per the same area. And again, the prevalence was not as high, but it was of a similar level. We did a further study on a further population of wood mice. This time we did a spatial and genotypic distribution. So if our hypothesis is correct that the parasite has been transmitted vertically from mother to offspring, we would expect the parasite to be transmitted through families and not to be distributed evenly in a geographical location. So we genotyped the mice, we tested them for toxoplasma, and we looked at their geographical locations. We looked at the populations of mice. We found there were 4 genotypes, the red genotype, which is split into 2 populations, the green and the blue. And what we found was that in those genotypic populations, there were different prevalences of infection. And when we did the statistical analysis on that, we found there was a highly significant association between genetic group of mice and there was no association with where we caught them. So again, we suggest this is evidence to suggest that perhaps the parasite has been transmitted vertically in these mice. We want to look at sheep as well. Sheep are really important in the UK. Toxoplasma is responsible for a lot of lamb losses, and we were interested in asking this question about vertical transmission in sheep. And the background to this really is that vertical transmission is not important in sheep. That's the current thinking, and that sheep are generally infected by infecting bedding and water. And also, it's believed that immunity occurs following infection, and that lifelong immunity occurs. So we wanted to sort of look at this particular issue. In terms of my overall interest of the parasite transmission, one of the things that it does by looking at a herbivore is it reduces one of the routes of transmission, the route of carnivory. So that helps us to begin to hone in on what might be causing the infection. So we used PCR to detect parasites from newborn lambs. We wanted to use a non-destructive technique because we wanted to explore a natural population. So we sampled umbilical cord tissue, which we found to be a good indicator when we compared live lambs to aborted lambs. If we compared the umbilical cord from an aborted lamb to its internal brain tissues, we got a very good agreement between the umbilical cord and the internal infection. And when we looked at a large number of lambings, we looked at a total of 392 pregnancies, and we scored them as successful pregnancies and unsuccessful. And an unsuccessful pregnancy was one where at least one of the lambs was aborted or born dead. And what we found was that, interestingly, in the successful pregnancies where there was no abortion, 65% of the lambs we detected parasite DNA present in there. So these lambs would have gone on to become healthy sheep. In the unsuccessful pregnancies, we found a higher percentage, and overall in the flock, we found 69%. So this is very high compared to previous data that has been published and compared to the kind of general feeling. So one of the things that we think is happening here is that there's abortion associated with infection, but also there are a very large number of healthy lambs produced from this vertical transmission. So if that's the case, then we would expect to be able to track the infection through generations of sheep. So what we looked at was we looked at families of sheep on a particular farm. So if we have two hypotheses, if the hypothesis is that the sheep are infected by oocysts in the bedding, okay, we would have an expectation that the infection should be randomly distributed across different families of sheep. Okay. If, on the other hand, we had vertical transmission from ewe to lamb, we would expect to see different infection levels in different families of sheep. And this is the data, and we looked at a pedigree flock where we could track the history of this flock going back 11 years, and we were able to sample for toxoplasma in the lambs over a 3-year period. And these are the data that we've got. And so this is looking, first of all, at abortion in different families. So each of the lines along here, there's 2 columns, each of the lines is a family. And we found that there were some families with high levels of abortion, so 48%, 40% are the ones in the red circle there. And on the same farm, families with low levels of abortion, so the ones in the yellow circle there, 0%, 5%, and so on. And we found a range of different abortion levels. When we looked at infection with toxoplasma, again, we found some families with very high levels of infection. 100% of the lambs we tested were infected, and in other families, we found 0. And when we looked at both, we found, again, families with high levels of abortion, high levels of infection, and other families with low levels of abortion and low levels of infection, and a range in between. And we found a very highly significant correlation between percentage of infection and percentage of abortion. And here's some examples. This is a family with a high frequency of abortion and infection. You can see all the red circles indicate lambs that were aborted in this family over an 11-year period. This is a family with an intermediate level of infection and abortion, and a family here with low levels of infection and abortion. These are all families of sheep on exactly the same farm with the same sort of surroundings and husbandry. So we suggest that, and that these are all significant differences in terms of the randomness that we would have expected from our hypothesis. So we think this is strong evidence for vertical transmission in these sheep, and we actually think that breeding from infected families may increase the infection and abortion rates. And we looked at the risk of this, and we calculated that breeding from ewes that have previously been shown to have abortion and infection have a 55% subsequent risk of abortion and a 69% subsequent risk of infection. So this is quite contrary to the current thinking, and in fact influences the advice to farmers. So what about humans? So what we wanted to do is we wanted to do a similar study in humans. We wanted to look at umbilical cord tissue from humans to see if we could investigate this question in the same way. So again, the current views, typically congenital transmission occurs at very low levels, 1 to 2 per 1,000 live births. It's rare but can cause significant disease, and serological systems are those that are most commonly used to look at human infection, and that may be, as we'll see in a minute, one of the things that is different between our data and other data. So we did 2 studies. One involved a hospital in Libya where we collected 276 umbilical cord samples spanning 272 pregnancies, and a study in a hospital in the UK in Manchester where we got 94 umbilical cord samples. In the Libyan study, we found that 27 out of the 272 babies were infected with toxoplasma using the PCR detection system, so that's about 10% infected. Fortunately, there were very few unsuccessful pregnancies, but there was no association in this case with toxoplasma and the unsuccessful pregnancies. The case in Manchester was even more worrying, where we found 1 in 3 of the umbilical cord samples taken from babies during birth were infected with toxoplasma. So again, this suggests to us that there's really quite a high frequency of perhaps congenital transmission, and this may produce healthy but infected babies, and there may be no future problems associated with. So we suggest this is possible evidence for vertical transmission in humans as well. So just to come back to the life cycle, we've got this complicated life cycle. And what we're proposing is that third transmission route might be much more important than we previously think. We're not saying that animals and humans, the marine mammals, etc., are not seeded with infection by oocysts from the cat, but we believe that maybe vertical transmission may be propagating the parasite in a more frequent manner than we previously thought. And our data are consistent with these transmission cycles that bypass the cat, and we would suggest that vertical transmission may be a possible explanation. I'd just like to acknowledge collaborators. Lots of people in our lab in Salford doing this work, people at the hospital at Manchester and in Libya, and our colleagues in China, Professor Zhilong Shen, Xiaorong Lun, and Sharon Hughes, who maintains the closed colony of mice that we worked at. Thank you very much.
Questions? No.
Thank you very much, Professor Hyde. Give us such a wonderful presentation, and it's very important for us to know the vertical transmission, the significance of the vertical transmission in the prevalence of toxoplasma infection across the world. So very interesting. And my question is, you found that the circulation of toxoplasma infection may occur in sheep and the rodents without the final host of cat, right?
Yeah.
And it has a very strong genetic background.
Yeah.
The family-based research. And you know, based on our investigation, I mean serological investigation in humans, we, just as I mentioned, that there is about 5 to 7% of serological prevalence in China. We also found some seropositive pregnant women, and we followed up their offsprings, the babies, and then detect the antibodies in the babies. And also we collect the placenta tissues and aborted the tissues, and then do PCR to try to identify the vertical transmission. Unfortunately, few cases were found through the transplacenta infection. So I want to know, is it comparative of this animal experiment to the human infection?
I don't know the answer to that question. It's very clear in humans that certainly the vertical transmission that we measure is much lower than in animals. So in sheep we get 69%, in humans 10%, 30%, depending on the study that we've looked at. So it clearly is much lower in humans than it is in the animals. We're not sure whether PCR is a more sensitive technique than serology. One of my hypotheses is that actually if the parasite is transmitted to the fetus prior to the full development of the immune system, it may be that those individuals, those newborns, are not detected as seropositive. Now, I'm not sure if that's true or not, but that's one of the hypotheses I have. I think whether the human is a good animal, whether the animal models are a good representative of the human system, I don't know either as well, but that's something I think we need to explore further.
Anyway, it's a new concept about it.
Yeah. New and very controversial, and not everybody believes it.
Thank you.
Okay.
Thank you very much for your excellent presentation. In this model of vertical transmission, what happened with the immunological system of the host? There are some coevolution between
the
host
serology
and
no. In most of these studies we didn't do serology. We were only using PCR, so that's a possible confounding issue. With some of the samples, we've done a comparison of serology with the PCR, and we don't find very good agreement between the two methods. Now, we're not sure what that says, and that may also provide some of the reasons, the controversies, I guess, as to whether this particular scenario is happening.
Thank you very much.
Only one more question.
Okay, quick one. So when you said you don't find any good correlation between serology and PCR, so which one is more sensitive? And how do you explain this lack of consistency?
I think the answer is not about sensitivity, but it's about what they're measuring. So serology is measuring obviously exposure to infection, but it's also I guess See, one of the things I'm concerned about is that newborns in particular may well be, if the immune system has developed to either not recognize the parasite or to recognize it as self, it won't produce antibodies, and so they'll appear seronegative. Now, I haven't found any data on that to either support that or to refute that, but that's something that I think is important to explore in the future. So there may be a discrepancy there because we're working on newborns. And of course, DNA only detects PCR only detects parasite DNA, not living parasites, and so there are a number of reasons why the data might be discrepant, and I guess the challenges are to try to tie the two things together.
Thanks, Dr. Hat. speaker is Dr. Hanan Kater from Benya University, Egypt.
What's called presentation? No. No. It is there. I'm sure it is there. Symbiotic. This is this one. Hello, everybody. Actually, speaking behind the podium, look, the flow of energy. That's why I'm going to speak away from the podium, and because what comes from heart goes to heart. I'm Dr. Hanan Kater, Professor of Parasitology at Banha University, Egypt. I'm currently working at the School of Biological Sciences at Bristol University in the United Kingdom. Today, I'm going to present comparative efficacy of symbiotic and declazuril on the performance of broilers infected with Eimeria acerulina in chicken. My co-authors, Dr. Ali and me, Hanan Kater, Shakir Siddiq, Mainada, and Kadakaria Morgan. Actually, when chicks start to ingest feed, they are very susceptible to pathogenic microorganisms, especially coccidiosis, which is a worldwide disease. Avian coccidiosis caused by Eimeria species, which is the most economically important disease of poultry, and it causes global economic losses estimated by over $3,000 billion annually. Coccidiosis is a serious threat to poultry production because Eimeria species survive environmental challenges and emergence of drug resistance. In modern poultry production, antibiotics are often used as feed additives. Why? They are not chicken feed. Because they suppress or eliminate harmful organisms from the intestine and to improve growth and feed efficacy. Of course, there is some disadvantages. Formation of resistance against pathogenic bacteria to human. Therefore, in the year 2026, there is a ban of the use of antibiotic as growth promoters in animal production by the European Union. Consequently, researchers and nutritionists are searching other alternatives for improvement of the performance of broiler chicken. For example, symbiotic. Broiler chickens are chickens that produce for meat. We always hear the expression of probiotic and prebiotic, and they are essential for good digestion. But what is the difference between them? Actually, prebiotic are non-living, non-digestible special form of fiber or carbohydrates. Whereas probiotic, they are live active microorganisms that when administered in adequate dose, they will have a beneficial effect on its host. Prebiotic can survive heat, cold, and acids, but probiotic are more fragile, vulnerable to heat, and they may be killed. Prebiotic perform their role by nourishing the bacteria that live in the intestine, whereas probiotic fight the harmful bacterial species present in the gut. Here, the probiotic is the bacteria, and the prebiotic is the food for the bacteria. Therefore, symbiotic, the expression symbiotic means together. Symbiotic means pairing a probiotic with its preferred nutrient, prebiotic, which allows probiotic to stay present in the body for a longer period. And it's an efficient way to maintain all the health benefits they confer. So our aim of the work, using symbiotic for control of experimental infection with Eimeria cerevisiae in broilers, and study the effect of symbiotic on growth performance and some biochemical parameters of broilers treated with or without diclazuril. We use diclazuril and symbiotic, which consists of probiotic, which is Bacillus subtilis, and prebiotic, which is lactose, as 1 kilogram ton per feed. Our experimental infection, on the 8th day of age, we infected birds, it's 1-day-old chicks, 150 one-day-old chicks with sporulated oocyst of Eimeria cerevisiae, directly intra-group through a stomach tube. Here the experimental grouping. We divided the birds into 5 equal groups. Each group consists of 30 birds. The first group served as the negative control group, which is non-infected, non-treated. The second group, which is the positive control group, which is infected and not treated. The third group is infected and treated with diclazuril. The fourth group is infected and treated with symbiotic. The fifth group is infected and treated with both diclazuril and symbiotic, and I'm going to call it the mixed treated groups. And I'm going to call the fourth and the fifth groups are the symbiotic treated groups. Okay? So, and here is the timetable. Here the, starting from age 1 till 42, which is 6 weeks experiment. On the 8th day, we made the experimental infection. On the 5th and 14 days, we made coprological examinations. 7th and 14th days, the effect on growth performance, as we going to see. We got very, very interesting results, actually. Regarding reduction of oocyst production, treatments significantly reduced oocyst production 70 and 75%, while in the mixed treated group, there was superior production of oocyst, 89%. Lesion scoring has been improved in all treatments due to improvement of the intestinal morphology. As you can see, improvement of the lesion scoring. Regarding growth performance, symbiotic treated groups, the last group, there is a significant increase in the final body weight, final cumulative body weight gain, and reduction of the feed conversion ratio, which is great for producers, actually. Regarding biochemical analysis, there, in the positive control group that's infected and not treated, there is a significant decrease in the protein, total protein, serum albumin, and serum globulin. At the opposite, there is an increase of these parameters in the symbiotic treated groups. Regarding lipid profile, the lipid profile has been decreased in the negative control group, and the good and bad cholesterol are decreased, everything are decreased. Whereas in the symbiotic treated groups, there is a decrease only of the bad cholesterol, as you see over here, total cholesterol, triglycerides, low density lipoprotein, whereas there is an increase of the good cholesterol, which is high density lipoprotein. To test the safety of our treatments, the liver function indicated by ALT and ST, and the uric acid and creatinine indicating the kidney function, and all treatments are safe for the chicken regarding kidney and liver functions. What about the mode of action of symbiotic? Actually, probiotic secrete antiviral, antibacterial, and antifungal chemicals. They form a physical barrier, actually, which hinder pathogenic microorganisms. Any pathogenic microorganism, it could be bacteria, it could be coccidia, whatever. Actually, they also create acidic environment which promote absorption of iron and other minerals. In more details, probiotic, these are the bacteria, enhance development of mucosal barrier, enhance immune system, and synthesis of vitamins. Also, they lower fat, the lipid profile, as I mentioned before, because they affect the bile acid metabolism. They produce short-chain fatty acids, and these short-chain fatty acids reduce they lower the pH in the large bowel, which is very important. I'm going to discuss that later. Last year, there was a publication of mine in the British Poultry Science about using short-chain fatty acids, which butyric acid, on Eimeria maxima. It was published in the British Poultry Science. We got very interesting results. Here are the results from short-chain fatty acids. They decrease metabolic needs and increase the availability of nutrients to the host. They hamper pathogenic bacteria, lessen bacterial fermentation, which reduce toxic metabolites, facilitate beneficial bacteria, that's the probiotic, regulate tight junction protein, which maintain the mucosal barrier, improve protein digestibility and minerals. All these factors improve the health of the gastrointestinal tract and improve body weight gain and food conversion ratio that was explaining the results. Here is the citation of our previous work. So our conclusion, symbiotic not only control coccidial infection, they improve growth performance of broiler chickens, they increase the total protein albumin and globin, which reflected on the immunity level, and decrease total cholesterol. Other benefits of symbiotic, they give producers the opportunity to eliminate in-feed medication from their programs. Therefore, they increase the profitability on poultry production. And they provide people with healthy and nutritious poultry products. And they are friendly to the environment, producers, and consumers. So everybody's happy, as I am happy to present this work to you. So, symbiotic are useful supplements in broiler diets. They are alternative to growth promoters and antimicrobial drugs. And thank you, and don't miss my presentation tomorrow about we should be ahead of it. What is that? I'm going to tell you tomorrow. Thank you.
Thanks for your nice talk. Questions?
Any question or comments? Doesn't have to be a question. Could be a comment. You like my presentation, you don't like it, anything. Just say anything. What? Thank you, I appreciate you.
No questions? Yes, please.
This symbiotic increases also the fertility of the poultry? Increases the fertility? How is the fertility of the birds?
Actually, these birds are broilers.
Mm-hmm.
They are produced for meat. Their age is just 6 weeks. They are killed and they are not used for production. They are used for meat production. Their age only 6 weeks. They are these chickens are used for your chicken table meat. So they are not used for production actually. But it will affect fertility, of course, because it enhances the immunity. The general health are improved. Everything's improved for the best. And also it gives you very healthy and nutritious meat, which is low in cholesterol. If the cholesterol level in the serum is low, that reflects on the breast and the thigh cholesterol. And then you eat meat poultry with low cholesterol, which is very healthy, and very high in very high level in good cholesterol, and very low level in bad cholesterol. So everybody's happy, as I told you, and it also prevents cancer. It prevents cancer.
I have a question.
Okay, go ahead.
How about the use of probiotics in humans?
Actually, it's recommended to be used for human. There is supplement for the probiotic, but if you take probiotic, don't take it only probiotic. Take the food for the probiotic, for the bacteria, which is prebiotic. So if you eat yogurt, you should eat fibers with it, or lactose, or موز اللي هو OS monoinosaccharides.
Thank you.
Thank you.
Oh, one more
Any
question,
question?
please. The last one.
Thanks for your presentation. Is it expensive to produce these symbiotics? So they can be applied in industry. Okay. So it's also already used in industry. Oh, thank you.
And there is a lot of supplements with probiotic and prebiotic both for human use also. Okay? Thank you.
We have a change in the program. I'd like to introduce the next speaker is Dr. Jinlin Shen from Nantong University, China.
Jillian, thank you. Can I take it? Ready?
Could we get a microphone from the back, please? Handheld one, please. Thanks. Yeah.
Okay, can I open it?
His biography is on page 52.
Thank you. Hello, everyone. Okay, my name is Jillian Chen. You can call me Jillian. Okay, I'm associated professor at the Department of Pathogen Biology of Nantong University from China. Okay, today I'm going to present our study on the possible molecular mechanisms of schistosoma japonica soluble egg antigens restricts fibrosis. Liver fibrosis or scarring of the liver, okay, which is a very serious disease, which is a wound healing response that engage a range of cell types and mediators to encapsulate injury. Although acute injury can activate mechanisms of liver fibrogenesis, okay, the sustained signals of associated with chronic liver fibrosis induced by variety of etiological factors such like hepatitis B, C viruses, alcoholism, and schistosome infections are very important for liver fibrosis accumulated. Schistosomes are a group of closely related flukes that habit in the portovascular system. Of the 7 species that infect human, schistosoma japonica mainly found in China and Philippines. Okay. The secure infect the skin or penetrate the skin of the infected human and develop into adult world. Okay. Parasitizing in the mesenteric wings while the hepatic portal systems and cause schistosomiasis. Okay. Schistosomiasis is one of the most prevalent parasitic diseases and often results in chronic liver fibrosis, okay, occurred secondary to the inflammation granulosus. Although a large number of cells contribute to the liver fibrosis, okay, hepatic stellate cells are the key factors of liver fibrogenesis. Okay. Hepatic stellate cells can be activated, okay, express alpha smooth muscle actin, we call alpha SMA, and produce a large number of collagens. Okay. Activate stellate cells is stimulated further by key cytokines into myofibroblast. Okay. In the past, majority of researchers think that liver fibrosis is irreversible. Okay. But now, increased evidence suggests that reversible. Okay. As the key effector cells, hepatic stellate cells, HSCs, might return to quiescent-like state or undergo apoptosis, okay, thereby reversing liver fibrosis. Okay. In our previous studies, Schuster-Schumann Japan in fact optimized for 6 weeks, 12 weeks, even 24 weeks. Okay. We detect two important proteins. The first one is Destiny, the green one, okay, which is a characteristic marker of HSCs. Okay. Another one is green, PPAR gamma, peroxine proliferate activity receptor gamma, okay, which is one of the most mediator, okay, which is highly expressed in the quiescent HSCs, but it is downregulated significant during the development of HSC activation. Okay. We found the numbers of Destiny and PPAR gamma double positive cells decreased in 20 weeks post-infection, but increased at 24 weeks. Okay. So what does it happen? Okay. It means in the process of the infection of the Schuster-Schumann Japonicule, active soluble antigens from the Schuster-Schumann Japonicule secreted by miracidium from the Schuster-Schumann Japonicule and released through the shear of X of Schuster-Schumann SEA could reduce. It is one kind of cell line of human hepatic stellate cells. We found SEA, the treatment of SEA, could inhibit the proliferation of HSCs and induce quiescent-like state. Okay. Additionally, the treatment of SEA also provoke cell cycle arrest at G1 phase. Okay. Furthermore, SEA could inhibit activation of HSCs while decreasing of SMA and increase PPAR gamma expression. Then we isolated the primary hepatic stellate cells from the normal mice and the Schuster-Schumann Japonicule infected mice. Okay. We found SEA did inhibit the activation of HSCs derived from cell lines such like AX2 cell and primary hepatic stellate cells and the activate primary HSCs, thereby reversing liver fibrosis. Okay. So we have idea, or our data demonstrated that the activated HSCs might return to quiescent-like state. Okay. Besides that, some researchers found that SEA could harbor the protector to undergo apoptosis. Okay. So we attempted to explore whether SEA, the treatment of SEA, could induce apoptosis of activate HSCs. Okay. We found case-based activated and expression of cleavage caspase 3 and 8 are downregulated significantly. Okay. Then we use ZVAD FMIK caspase inhibitor. We found the elevated expression of cleavage caspase 3 and 8 reversed. Okay. So we know that the SEA from the Schistosoma japonica did cause apoptosis, but which molecule or protein can mediate the SEA-induced apoptosis? Okay. Based on some study that several molecules can be reported expressed by HSCS, okay, such like DAS receptor 5 and DAS receptor 4. Okay, we found the treatment of SEA could reduce the expression of DR5 instead of DR4. In order to confirm it further, we use DR5 SHRM, suppressing the expression of DR5. Okay, the elevated expression of caspase 3 and caspase 8 are partially reversed. Okay. As an important tumor suppressor, P53 is involved in the cell cycle and apoptosis. Okay. We detected the expression of P53. We found it is increased by the treatment of SEA. Then we use SI-P53, okay, is transfected into LX2 cells. Okay. We found the expression of DR5 and caspase 3 are significantly reduced. Okay. And the nucleus communication of P53 would be detected upon to the stimulation of SEA. Okay. PI3K and AKT are very important pathway for suppressing apoptosis. Among them, AKT is the most important mediators. Okay. So we found the treatment of SEA did decrease the AKT. Okay. So we use PPAR gamma agonist GW to increase the expression of AKT. We found, okay, the elevated elevated expression of cleavage P53 and DR5 reversed significantly. Okay. So we found SEA from the Schistosoma japonica had the effect on AKT phosphorylation. Okay. Then increased the P53, then work on the DAS receptor 5, then increase the caspase-related proteins. Okay. Subsequently, cause apoptosis. Okay. Thereby reverting liver fibrosis. Okay. Okay. In a word, in the infection of the Schistosoma japonica can activate HSCS. At the same time, activate HSCM might undergo quiescent transformation, and sometimes they will undergo apoptosis. Okay. But is there any other ways to reverse fibrosis? We are on the way. Okay. So our works are supervised by Professor Duan. Okay. So if you have chance to go to China, welcome our lab. Although my boss is not so handsome. Okay. Never mind. Okay. There are some part of articles that from our lab. Okay. Thanks for your attention. Okay. Any questions? Okay.
Nice talk, Dr. Chen.
Oh, sorry. Thank you. Thank you.
Thank you very much for your presentation. In my knowledge, the SEA induced the fibrosis in liver, but your story is just, inhibit the fibrosis. I believe your story. Also, I believe my knowledge. So I think the SEA has two sides. One side induce the fibrosis, another side it can attenuate the fibrosis. Maybe it inverted in the quantity. If the SEA express over, they can express the fibrosis. If you only say the SEA can attenuate the fibrosis, I don't think it's okay. Yeah, I think they are involved in the quantity of the SEA.
Yeah, I have think about these questions. Okay, I think it is a balance. Understand? Yeah, sometimes the infection of schistosoma japonica can cause liver fibrosis, but at another side, okay, in China we call balances is the very important things. So in our studies, we found SEA have another function about the liver fibrosis. Okay, but so up to now, our data contaminated or suggested that SEA from the schistosoma japonica had this function about reverse fibrosis. Okay, we are on the way. Okay, thanks for your questions.
More questions?
This is a very interesting story.
Thank you.
Just as mentioned by Professor Lee. And does your story means the continuous deposition of X in the liver may alleviate the fibrosis? It means, the continuous production of X in the liver and reversely it reduced the severity of hepatic fibrosis. Is this true?
We have a long way to go. If you say it is did reduce reversion of the liver fibrosis, as we are a long way to go. Okay, so just for our data, we suggested that it is the truth. But we don't know in the future because we have a long way to go. Maybe sometime in the future, this SEA could or harbor the potential to reverse liver fibrosis. Who knows? Because science is the miracle. Okay, maybe in the future we use this SEA to treat liver fibrosis. We have no idea about that thing. Okay, thank you.
Toxins against toxins.
Thank you.
No more questions? Thanks, doctor.
Okay, good afternoon, everybody, and thank you for coming back to this afternoon's session. I'd like to introduce our first speaker, who is Professor Tingbao Yang. He's from the Sun Yat-sen University at Guangzhou in China.
Okay, hello, everybody. Good afternoon. Yeah, after lunch, I suppose most of you, all of us are tired. And for me, and because I came here yesterday afternoon, so a little bit tired because of the jet lag. So I'm so happy to have the opportunities to present my the short story about the phylogeographic studies on Angiostrongylos cantonensis and its intermediate hosts, Achinetta follicule. So that's it also called African giant African snail. Phylogeography has been more than about 30 years here, when the concept was first. I short already. Yeah, and this concept was proposed by Avis, and that means they want to find the relationship between the genealogy and the biogeography, because when the animals plants, animals particularly, when it leaves its original location and it moves to very far away, and the separation between them and between the genetic comparison with the origin area, and they will have difference. They accumulate difference. So they want to identify it's a kind of intraspecific evolution process based on the present phylogeographic pattern. And later, because of a lot of molecular data are available. So, and a lot of molecular markers are used for this purpose. And the general objective is we're trying to find the process from the intraspecific microevolution to interspecific macroevolution. That's the so until now, already some phylogeographic model has been found for the terrestrial, but for marine environment, that's different because there's no real barrier between different environments. But there has been 3 patterns has been found, and the first one is that they in different locations they have different. Some of them with inconspicuous genetic structures, and for this one it's in different connected environments, but they are with very obvious genetic divergences. And the third one is the locations isolated, and then they have very clear, large divergences in their genetic structure. That's the 3 basic patterns have been found. And here I would like to give a little bit about the introduction for why I started to do this. As most of us know, Angiostrongylos cantonensis, it is also called the red non-worm, and it becomes a worldwide particularly in the tropical area. In China, in 2006 and 2009, and in Beijing and Wenzhou, and many people, more than 100 people, have been infected. They infected with this worm and get very serious disease. And there are some studies based on mitochondrial DNA sequence, and they found these parasites are multiple origin. And there are several studies in China. They have the similar, but their studies based on. This is what they have done. Yeah, the results. But I found their studies are based on several samples for each locations. For example, many locations, they only one or two samples. But we thought, we'll think about if you want to use only one or two samples to represent the genetic structures, it's very dangerous because only the sample is too few. So that's why, because this parasite, this nematodes was found and described by a professor in San Yat-sen University. So that's about 2 years ago. We thought about we should do something about how the parasites and migrates and to different area, and if it is in the south part of China, where is the origin of these parasites? And also, there are some suggestions about the origin. They said the Southeast Asian countries are the origin of these nematodes, but we are not sure. So we want to do some research. The objective of the present study is going to be we want to evaluate the. We use different genes because based on different gene sequences, and you may get different results. So we choose ND2 and SAB for both the nematodes and the hosts and the slayer, because based on the investigation and the reference, we found this slayer now it is the main intermediate hosts. Some, for example, golden apple, and actually that slayer is normally in the water. So it can, many species of slayers could be the intermediate hosts. But we found these snails is the commonly infected and with the very high prevalence and abundance. So we want to find if they are the same pattern, if there is the same pattern for the slayers and the nematodes. And we want to assess the genetic homology of this nematode and its hosts in South China area, because this slayers was introduced to south part of China only about 80 years ago, and before there's no record. There were no records for these snails. And further, we want to elucidate the population genetic structure of these nematodes and its hosts in the south part of China. And we want to find, we are trying to find the migration routes of these nematodes. And this is the snail we sampled, yeah. And this is a distribution, because this snail is actually is a tropical ones. And originally, it is called a giant African snail, and it is from the originally it is from Central Africa, yeah, from Africa. So it doesn't goes to the lost part, and we made the investigation, and even in the lost part of Guangdong, and we couldn't find these snails. But it couldn't climb over the high mountain. This is a distribution, and this is the third stage larvae of the nematodes. And we sampled, in each position, we sampled a lot of snails, because from each snails we only collect one individual, only one individual of nematodes, so that in the area we can get more nematodes, and so that the data will be more representative. And this is the location we sampled in this area, and we found in Hainan Island and in Yunnan province, because their literature, it said that this is the origin of this area is the origin of the nematodes. So Guangxi and in Guangxi and in Guangdong, Taiwan, and Fujian. The Fujian also, because they are snails, they say this snails was introduced from Taiwan. From Taiwan. In Taiwan, we choose four locations: one in the east and west and the south and north. Yeah, Taipei. That's the general process we did. This is the primers we used for the nematodes and for the snails. And this is the software we used. I don't want to. Oh, okay. Now this is the results. Because based on SAT-B and ND2, we got the. We didn't show all the data, but we only want to show because based this haplotype diversity, and it will show you the genetic diversity. And from here, we found in Yunnan province, based on ND2, actually both the ND2 and the SAT-B, and we found the Hekou in Yunnan province. Yeah, well, I said some people said, based on the investigation, morphological study, and somebody said that area should be the origin of the nematodes. But here we found in this area, and the genetic diversity is the highest. Both Pearl and Hekou, both of these are in Yunnan province. Oh, sorry, this is the snail. I'm sorry. Yeah, this is the snail. Yeah, and this is the phylogenetic analysis based on the sequences. But just to show you, different color represent different area, and we can find. We can find, this is, this area is in Taiwan, in Taiwan. South part of Taiwan. And so we can find that there are two divergent branches, and one based on two genes. And the star-shaped structure and may imply the existence of population expansion, because the snails, the population was deduced to experience historical demographic expansion. And this is also the about the genetic structure of the snails. But we found there are genetic structures because based on the statistic, and we found it is with the because it is very different because we suppose there's freedom, but actually there are very significant difference from the p-value. We found it is different from the from our hypothesis. That means there are certain percentages of variation from among different among populations. And Tajima-D and the Fuchs FS tests also revealed the existence of genetic structure, and also it means the population has experienced historical demographic expansion. And this is the results about the snails. And here is the snail, because we want to compare and if they are similar or of the hosts and the parasites. No similarity or not. And here we found Herco also based on 2 genes, and also Herco is with in Herco area and the population with very high genetic diversity. Here it is higher than other areas. And this is the phylogeographic relationship of the nematodes. And here we found Herco, the samples from Herco, it can become clades in the root, in the base of the trees. We can find one here. And a few of them, few of samples in Herco also appear in other. For this one, these are from the Guangzhou and from other parts. But Herco can form one independent, clades. Here we use some data from Thailand. We included some more gene data, scientific data from Thailand. And we found that Thailand, they joined to this group, but the Herco group also exists here. So we found maybe we suppose Herco is still the origin. This is the when we put in the location, we found. Oh, sorry, that must be ND2 because I modified. But here we also found the in Herco also it becomes in the root, and we found one clades, mainly composed of samples from Herco. So generally, we think Taiwan and those samples from Taiwan and Thailand, they get together, and this is the significant exact test between different populations, and also but we couldn't find the that most of them are not significant. That means we couldn't find population structure, clear population structures. And here we can find except the Hekou, except that from Hekou, and the Hekou population with all other population together, and it account for 67 point of the difference of the variation, but among population others only can explain a very low proportion of the variation. That means more than 2/3 of the variation from between Hekou population and the other population as a whole. That's the main result. Yeah, among different populations, that's variation over 80. And here we also make Tajima-D and this test. We found there's no significance. There's no much different. That means this actually this parasite the nematodes doesn't have very clear genetic structures. So it's a short story. It's a short story because we are sampling. I have some other samples from Thailand, and from now we are doing the sequencing. Yeah, so I haven't included them. So it's an ongoing research. So, and we think this parasite, this snail, population in Taiwan generally have higher genetic diversities. That's what we found. And in Taiwan, we detect historic population expansion. That's the snail itself. And because this snail was first, just not long ago, based on reference, the literature, and this snail was recorded in Taiwan very long ago. But in mainland China, in south part of China, only a short time. And it was introduced from Xiamen. So when this parasite in Taiwan, it has a process, it experienced the population increase, expansion. And also in south part of China, and the snail in Hekou might be artificially introduced or migrate naturally from southeast country, because this snail was introduced to Taiwan from Southeast Asian countries. So, but from Yunnan is bordered with border on Vietnam, so it could migrate from Vietnam to Yunnan, and then gradually, and it may migrate to the eastwards to south part of China. And this population in Taiwan was and is composed by two slightly differentiated groups, and with genetic differentiation. And but it's a lot of it is not a random mating population, and the snail might be introduced from different. Yeah, we just think about that. The snail might be introduced from different areas, different sources, and one of which was introduced to China, and the other part, because in Taiwan we know the central part is high mountain, the east part and the west part, and also it has been different strains of nematodes already recognized and described, and they have very clear genetic difference. So it could be. Oh, sorry. I think the separation, could be. Yeah, but it's very complicated. It's very difficult to explain that. As for the nematodes, nematode, the origin of nematodes and Hekou, and based on different genes, Hekou, Yunnan province might be the origin, or at least one of the origins of Angiostrongylos catanensis. And because they have very obvious high gene diversity than other areas, it was reflected on the geographic relationship on the tree. And because we only sampled about 30, and 30 of the haplotype from Hekou, from the one clade, only 30 samples. And also before Jones already he suggested the Southeast Asian country are the origin of this snail, because the Hekou is brought on there. And also these guys in Japan, and they proposed, because that's what I showed in the introduction, and they proposed Angiostrongylos catanensis was a multiple origin, based on the samples from different area, including south part of China. But some of them, their samples are too few. But actually, I can't trust these results. And the summary, the results, and our present results, it shows that the snail is a non-random mating population but without significant structure. But population experience demographic expansion. For the parasites, just the Hekou is the we think because of its higher genetic diversity, we think it is the origin, and but it doesn't experience demographic expansion, because we think about that, because this parasite, these nematodes, it has more than 10 species of snails, and rats could be their hosts. So it's very complicated. And when we just studied one snails, so the role of one snail, I think they take very little part in the spread of these nematodes. And now we think, although this snail is very important, and based on the prevalence and the intensity in the spread of these nematodes, but it is still very hard to see how important of these snails in the spread of the nematodes. This is the result. Thank you.
Great, thank you very much. I'm afraid we're running very far behind time now, so we won't have any questions. But if you've got questions for Professor Yang, please feel free to ask them during the coffee breaks later. Thank you very much.
Sorry about that. Okay. The next speaker is Dr. Ahmet Ozer from the Sinop University, Turkey.
Good afternoon, everyone. I have a presentation here with the title of Assessment of Endemic Tooth Carp and Invasive Mosquito Fish. health by means of relative condition factor under the co-infection of different parasite groups and revelation of the effects on their competitive interactions. Fish serve as hosts to ecto and endoparasites that exhibit a wide variety of life cycle strategies. Parasites can reduce growth, fecundity, and survival, and change behavior and sexual characteristics of the infected fish hosts. The host-parasite relationship is often unbalanced, and parasites can severely compromise the health of their host and even kill them. Most of the studies showing the effects of parasites on wild fish populations have been carried out in freshwater ecosystems, and parasites on their host fishes reported to be causing either direct mortality or indirect effects such as decline in recruitment, success and growth, alteration on host behavior, reduction in swimming speed, and increased risk of predation. However, the effects of parasites on the health status of endemic and introduced fish populations, the relationship between parasitism, condition, reproduction, and natural mortality of these species remains largely unknown, and as a consequence, parasites are often overlooked in fish health assessment. Studies on the effects of parasites on the health conditions of fish are very rare. Fish condition can be extremely important to fisheries management, as plumfish may be indicator of favorable environmental conditions, whereas thin fish may indicate less favorable environmental conditions. The relative condition factor, one of the commonly applied condition factors used to determine the effects of parasites on their host in natural populations, corresponds to the ratio between observed and theoretically expected weight for a given length and allows statistical comparison between the estimated value of key n and the central value of key n equals 1. The relative condition factor key n greater than 1 is indicative of the general well-being of fish, whereas its value less than 1 indicates that fish is not in good condition. So we have examined two fish species here. One of them is Gambusia halbrooki. It's a species of freshwater fish and is a member of family Poecilidae of order Cyprinodontiformes. They feed readily on the aquatic larval and pupal stages of mosquitoes and are called as eastern mosquito fish. It is also thought that this fish has potential to cause damage to endemic fish and other endemic aquatic life. On the other hand, Alphanius stamfordi is also a species of freshwater fish and is a member of family Cyprinodontidae of order Cyprinodontiformes and is called as killifish. This fish is fed on crustaceans, insect larvae, and some plankton species and has a potential of controlling mosquitoes. In this map, you see the distribution of both species. As you see in figure 3, Gambusia halbrooki is worldwide distributed. On the other hand, our endemic species Alphanius stamfordi is located only in this area located by the Black Sea. Fish from Lower Kızılırmak Delta were collected by electroshock device in the period between December 1910 and November 1911. We measured whole weight and total length. All fish underwent a systematic external and internal examination in tissues, including skin, fin, gills, eyes, intestine, so on. Parasites recovered were fixed and preserved using methods commonly applied, and our identified parasites are grouped as protozoa, monogenia, digenia, acanthocephala, copepoda, cestoda, and nematoda, and used for comparing the effects on fish health status at higher taxonomic level. Calculation of infection prevalence, mean intensity, and mean abundance follow the Bush et al. And briefly, the prevalence is the percentage of infected fish, while mean intensity is the average number of parasites in the total number of infected fish, and the abundance is the average number of parasites in the total number of examined fish. So you see the location in the map again. We used a formula that the relationship between the measurements of length and weight of fish was modeled by means of parabolic equations done by Froese 2006. I will go through these parts because it has so much details, statistical details. Only one thing, that the relative condition factor, Kn is the theoretically expected weight for a given length was calculated by using the estimated weight-length relationship curve. Then the relative condition factor corresponds to the ratio of observed weight and expected weight as below. So I'll go through these parts again. And we search for season, effects of season, number of parasite groups, and their interactions. Here are the results that we found on both fish species. So you see that endemic Aphanus stamfordi had 18 different parasite species. On the other hand, introduced Gambusia halbrooki has only 9 fish on 9 parasite species. So here are the details for the higher taxa that the prevalence in endemic fish was dominated by Digenia. And when we look at the mean intensity, it's for Aphanus, it's dominated by Protozoa. And it was the reverse for Gambusia. The others are less in prevalence and mean intensity values. Here are the photos of some parasites, and you see those parasites, and most of them are new records for Turkey, and some are also new host records. So I will just pass them by. These are trichotinids. There is one Vorticella, and on the right is Sebotriocephalus achilognati cestode. Here is monogenian parasite. We identified 2 different parasites. Here are the Digenians. You see different species here, and dominated as species diversity. And we have nematode here, Sproxis contortus, Hysteretilasium, and Schulmanella petruszewski. These are all from tooth carp. On the other hand, we see several parasites on eastern mosquito fish. Of course, they are less in number, and here are the photos of the parasites that we identified. The majority of trichodinid species are seldom involved in fish mortalities. However, trichodinids are known to proliferate massively on stressed fish and become highly pathogenic under these circumstances. Diagenetic trematodes, Postodiplastomum, Tylodelphus clavata, Diplostomum speciatum are the members of Diplostomid, and they do not have strict host specificity. Tylodelphus clavata and Diplostomum speciatum are associated with specific effects or non-specific side effects of parasites, including impairment of vision that leads to exophthalmos, cataract, and even complete collapse of the eye, which may be the cause of growth inhibition, death, and significant proportion of cultured fish. Acanthocephalans are thorny or spine-headed worms, which with aquatic life cycle, they fish as final or paratenic hosts and crustaceans as intermediate hosts. Here are some details that we measured, we calculated from our data that with parasites and without parasites. 0 shows without parasites and 1 shows with parasites. And for each taxa, you see Protozoa, Monogenia, Digenia-like. And from this data, we calculated the Kn relative condition factor. As you see, they are all equals on the right-hand side. They are all equals to 1 in our tooth carp, endemic fish species. There is no difference there. Here we see we did the same thing for eastern mosquito fish. We calculated weight and length. Then we again use the formula, and we saw, okay, Kn in most parts, Kn equals 1, but in some parts you see Kn is higher than 1, the estimated value. Especially in you see Monogenia, is the uninfected ones has higher. On the other hand, Digenia has infected ones has higher, and in cestode as well. So when we look at the combination or co-infection of the parasites, you see Digenia, only Digenian, and Digenian monogenian, Digenian protozoa, Digenian copepoda-like, and you see, this is in the tooth carp, you see most of them are equal to 1. Only there is one thing, Digenian acanthocephala, but the number of low the same data for eastern mosquito fish, yes, we see the similar result, Kn equals 1. So when we look at the influence of parasite load on the fish condition of the tooth carp, as you see, for tooth carp, we have 4 different groups co-infected at the same time. For eastern mosquito fish, we have only 3 groups co-infected at the same time. So you see 1, 2, 3, 4 in the figures showing the number of co-infected parasites. So here you see in the tooth carp, and similar to eastern mosquito fish, the Kn is generally more than 1. Here is the seasonal data for comparing both fish species, and we see there autumn and spring is one group like, which means Kn of Aphanius lower than Kn of Gambusia. And on the other hand, summer has the reverse and winter has equal data here. In overall, however, there is no difference between their relative condition factors. Here we see the seasonal data again, and for the conclusion, amongst observed parasite groups, Protozoa shows the highest abundance, which is resulted from more space and food on higher weight and larger length of fish, if it is considered notwithstandingly the existence of the other parasite groups. Amongst the observed parasite groups, Digenea shows the highest prevalence, which is resulted from more food intake of fish, if it is considered notwithstandingly the existence of other parasite groups. Direct impact of monogenian, acanthocephalan, copepoda, and cestode infections seem to be negligible due to very low level of infections. Clearly, co-infections with protozoan and nematode parasites, along with above-mentioned parasite groups, did not cause any decrease in the development of both fish species. Moreover, co-infection with protozoa and Digenea, having the highest prevalence and mean intensity, doesn't seem to cause damage to these fish, whose relative condition factor was equal to the expected value, which corresponds to the needs of both parasite groups. Seasonal influences of co-infections by all parasite groups were determined in autumn and spring on the condition of tooth carp. Parasitic effects in number, varying from 1 to 4, exhibited no adverse effect on the weight and in the mean condition as the number of parasites, infection increases. The lack of statistically significant negative impact of such parasitism may have been due to small number of infected specimens of both fish species. Interaction of season and number of parasite groups appear to be non-effective on the relative condition factor of both fish species. This study clearly revealed that endemic tooth carp has a well-balanced well-being despite 18 parasite species over its body, and similarly introduced east fish has also well-balanced well-being, and this situation faster than I planned. Sorry if something missed up. Yeah.
Thank you, doctor.
Well, thank you.
Some questions?
Yes, sir. Sorry? Why did you sample? I didn't hear. Yeah, fish, yeah? Yeah, let me go back. Here you see from the lower delta. It's located by the Black Sea in Turkey. Well, here you see. Yeah, it's this part. Just a coincidence that, I mean, this fish is distributed around here, and not inside the seawater. No, we collected them all in freshwater areas. All freshwater. Yeah, freshwater. But, of course, I mean. But not from the fish ponds. No. Okay, from the very small streams. Well, actually, this delta is very big, the biggest in Turkey. It's the hosting space for birds and all, so it's protected by the law. So it's huge, and it consists of more than 10 lakes. So everywhere.
Stability, stable. If the water, the environment is stable, and because I'm fish hydrology guy as well, so I understand because you separate it from different seasons. And also, in the fish, I concern. My question is about the stability of the water quality.
Yeah.
If you are from a very small stream.
No, everywhere. Actually, as I said, it's a huge area, and we sampled from different parts of there, not from small streams or something, huge lakes. But of course, we use shallow areas because these fish don't go deeper, and we detect them by only electrofishing because they are so tiny fish. Yeah. Yeah, very small. Yeah. Okay. Thank you very much. Thank you.
Thank you very much. Our next speaker is Dr. Mauro Cortes Velez from the University of Sao Paulo in Brazil.
Well, first, I would like to thank you, the organization, for the invitation and the opportunity to share with you some data of my work. So today I'm gonna talk about the leishmania, in particular the infection of a macrophage from a murine model. So leishmania has the ability to inhibit the macrophage response, and for that reason, this parasite can survive inside of macrophage. This talk is related with the function of the role of a CD200, that is a molecule that we try to understand how it works in the macrophage infection. So this is a cycle of a leishmania. Leishmania has three different forms. One that is released when the sandfly gives the blood meal. That is a metacyclic promastigote that is released in the skin of the host. Several phagocytes, especially macrophage, phagocyte this parasite, and the parasite has the ability, as I mentioned before, to survive inside of this macrophage. So the parasite get in, form a parasitophorus vehicle, and this parasite transform in another form that is a mastigote forms, that is the form that is gonna be in the host the entire infection. So this form start to proliferate inside of macrophage, and then the parasite get outside of the cell, infecting another phagocytes or another tissue, and then when another sandfly gets a blood meal, the parasite get into the gut of this sandfly and transform in the procyclic promastigote, that is the form that proliferate inside of a sandfly. So my talk is related with this form, mastigote forms, that is the form that is going to be in the host the entire infection. So this is the different forms of leishmaniasis. Leishmaniasis has the characteristic that is presented with different manifestation. So you can find a cutaneous form of the disease, the mucocutaneous form of the disease, visceral, and also the diffuse cutaneous form of the disease. The presentation of this infection depends on several factors.
included the genetic background of the host and also the leishmania, the type of leishmania that infected the host. This is a table I can show the different forms of the infection of leishmania. This is all the leishmania that we know so far, and it's spread in several countries around the world. So my talk is related with the leishmanias that produce the cutaneous form of the disease. And in my lab, we work with Leishmania amazonensis and also Leishmania major. The characteristic unique of Leishmania amazonensis is that this Leishmania can produce the cutaneous form of the disease and also the diffuse cutaneous. But it's not the only difference between Leishmania amazonensis and Leishmania major. Also, Leishmania amazonensis can proliferate inside of a macrophage, inside of the parasitophorus vacuole. Different of the Leishmania major that they proliferate inside of a vacuole, parasitophorus vacuole, but this is a small vacuole around the parasite. This is an image of immunofluorescence that I work a lot of in my lab. But the big difference between Leishmania amazonensis and Leishmania major is that if you infect mice from the background, the black background, C57 black, you can find this kind of graph if you quantify the lesion size of the footpad of mice. So you can see Leishmania amazonensis produce a huge lesion on the footpad of mice. It's completely different than the lesion induced by the Leishmania major, that in some point the mice can control the lesion in this background. So the big question is why Leishmania amazonensis is more virulent than Leishmania major. So in the beginning, we did a microarray assay, an infection of macrophage from bone marrow, and we infected with the Leishmania amazonensis 48 hours of infection. This analysis was made with my colleague Sho Hyun, and also all the process of the data was made with the Institute of Systems Biology, led with Alan Adler. So in that point, we got a lot of results, a lot of molecules for this kind of assay. But the more important molecule that we found in this analysis, in this data, is the detection of a CD200 in the macrophage infected with the Leishmania. For what reason? Because CD200 is a molecule that is in the membrane of different kind of cells. This molecule presents two domains of immunoglobulins, so for that reason belongs to the superfamily of immunoglobulins. And this picture presents how CD200 works. So this is, CD200 is expressed in some cells like neurons. And when attached to the receptor, CD200 receptor that is present in macrophage, this binding inhibits the function of macrophage. In some point, when happens an infection, in this case it's a viral infection, cells degrade this CD200, releasing the macrophage, and macrophage can be activated and control the infection in the process. So the main role of a macrophage is to control, main role of CD200 is control the macrophage function response, and also contain exaggerated immune response in different tissues. So our question was CD200 induction is important for Leishmania infection. Two things are very important here that I'm going to mention because for the number of my data, but the more important issue is that Leishmania control macrophage, no? And also to control macrophage, some particle release or expressed in parasite has to be ability to do that. So for that reason, we work with two different strains of mice. So we work with the wild type C57 black 6 mice and mice deficient in CD200. We did qPCR, Western blot analysis, and also infection in vitro and also in vivo. For you to show how we measure the lesion, here is a lesion induced by Leishmania amazonensis and compared with the control. And infection in vitro, we also we quantify the number of parasites inside of macrophage, and we follow on different time points. So to verify the results obtained in microarray, first we did a qPCR with the primers specific for the CD200. So in this figure, we look at the infection in different time points and the presence of the parasite and compared to the non-infected. So as you see here, 1 hour after infection, we got a lot of transcript of CD200, 1 hour of infection. This transcript came down and it started to coming up again 48 hours after infection. Here is immunoprecipitation where we can see then the presence of the Leishmania, we can see a detection of the protein and the macrophage infected. And the non-infected, we can see a small signal. And S is a splenocyte. That is a cell that we use as a control to express CD200. So in this point, we saw that CD200 is expressed early time points after infection of macrophage. After that, we try to look at the proliferation of this parasite inside of macrophage. So for that, we compared the wild type that is in black, and compared to the proliferation of macrophage that for deficient CD200. So as you see wild type, you can see proliferation or increasing number of parasites inside of macrophage that you cannot see in deficient CD200 mice. When we saw this result, we think that to add in soluble form CD200 to see what happened with this deficient mice. And as you see in the figure, when you add soluble CD200, you recover the phenotype of infection in this CD200 deficient macrophage from mice. One of a particular mechanism to control a parasite inside of macrophage is induction of iNOS and production of nitric oxide, as was mentioned in the early morning. So we look at the induction of iNOS, and as you see in 1 hour and 6 hours, you can see in CD200 knockout mice macrophage, they produce more iNOS compared to the wild type. And this happened just in the beginning of the infection. To confirm the releasing of nitric oxide in the supernatant of this infection, we quantify nitric oxide by the grease method. And as you can see, in 6, 18, 24 hours after infection, you can see a difference between a production of the nitric oxide and macrophage from a deficient CD200 mice compared to the wild type. That means that when you don't have a CD200 in the cells, you have a lot of iNOS production nitric oxide. To confirm the role of iNOS production in CD200, we use inhibitor of iNOS specific, that is LNMMA, that inhibit, not see any difference, but in the phenotype of CD200 knockout mice macrophage, you can see the recovery of the phenotype, showing the importance of the induction of CD200 and the role of also of iNOS response in macrophage. This is a picture, immunofluorescence picture that you can see the experiment before. This is a wild type with an inactive iNOS inhibitor. This is with the inhibitor that you don't see much difference because the system the parasite induce CD200, but in the CD200 deficient macrophage, you can see the recovering of this huge vacuole containing parasite. So our model is that parasite Leishmania amazonensis can invade a macrophage, induce the of CD200, and for that this molecule, the signaling pathway, can inhibit the iNOS, and for that the nitric oxide production. In knockout mice or macrophage, you don't have the CD200 molecule to inhibit this signaling pathway of production of nitric oxide, producing amount sufficient to control the proliferation of the parasite. What is the difference between Leishmania amazonensis and Leishmania major? For that, we did qPCR with the primers with the CD200 and compare both infection. 1 hour after infection, we got a lot of transcript of CD200, but it's not detected in infection with Leishmania major. So Leishmania major fails to induce this CD200 in macrophage. To confirm the result of the role of CD200 in the infection, we did infection of on the in vivo model. So for that, we use infection in mice, and we added we inject CD200 soluble in the mice to see here in green. You can see the lesion size growing up. When you add CD200 soluble in the mice, and when we inject it, we induce a bigger lesion. And the more important, when we infect mice with Leishmania major, we can see here the lesion that coming down. But when we add a CD200 soluble on the mice, we recover the infection of this Leishmania major. Also, we quantify the number of parasites in the lesion, and we detect a huge amount of parasite when we add CD200 in the model of Leishmania major. That usually too much parasite in the lesion. This is a graph to show the role of nitric oxide to contain the proliferation of Leishmania major. This parasite cannot induce CD200, but when you inhibit the infection of the Leishmania major in this model. So the Leishmania major fails to induce CD200 because they have the molecule or secrete the molecule that inhibit this path in the model of wild type or deficient mice. But when you add CD200, you can change the phenotype of infection of Leishmania major in the system. So this is my lab, all the people that work in my lab, and we try to understand right now what is important of the toll-like receptor and the induction of being shown to participate the process of infection of Leishmania, and we try to see if this pathway is related with the induction of CD200. So this work is made for my student Ismail Sauter. He worked with the different kind of deficient mice, especially MYD88 and TRIF knockout mice, and he started to do we saw in the first figure. This is the phenotype of infection of lesion formation of Leishmania amazonensis in wild type and MYD88 deficient mice. So you see, it's a bigger lesion more than the wild type. This is a lesion that you can see in the footpad of mice. The first result that we saw in this model is that in this macrophage from deficient mice, in MYDA8 and TRIF deficient mice, you cannot see the same signal of CD200 compared to the wild type. In immunoprecipitation, you don't have in macrophage. That work is starting, so he's doing more with what is the toll-like receptor important for that induction. Another thing that we try to see is compared to a cutaneous leishmania with that induce visceral form of the disease, and try to see if the role of CD200 in this kind of leishmania. That work is made for my student Albert Bresen with the same material. He saw that in infection with the Leishmania chagassi, a parasite can induce CD200, but the induction is in later point of infection. Here is 72 hours difference between wild type and not infected with the infected. And here the immunoprecipitation showing the signal of CD200 and the system. And also, macrophage is not the only cell that is participate in the immunobiology of Leishmania. Also, neutrophils has a very important role in the infection. And we try to see if CD200 also participate in induction or in the control of the infection or susceptibility or resistance of infection Leishmania. For that, we compared the induction of Leishmania chagassi in macrophage in neutrophil, and we saw that the signal in neutrophil is bigger than we saw in macrophage. Here is a spleen obtained from infection with the Leishmania chagassi, and we saw that the infection of this mice, we detect on the spleen a huge signal of CD200 in that model. We don't know if in the spleen that signal is from the lymphocyte, because lymphocyte also express CD200, but at some point he's gonna separate the different population of cells, try to see what is the cell important for that bigger signal on the spleen. Also, we're gonna work with I don't remember the name of the golden hamster. Thank you. And we're gonna see the role of CD200 in that model, because in the induction of the visceral form of the disease is more it's more easy to see in this model of a hamster. Also, the other thing that we try to see is if CD200 has a role in the difference between cutaneous form of the disease and the diffuse cutaneous form of the disease. For that, we are collaborating with Valeria, that they prepare this nice work related the form or the exposure of phosphatidylserine in the membrane of Leishmania related with the cutaneous and diffuse cutaneous form of the disease. For that, we got all these isolates and try to see the important role of CD200 in that model. I have time? No. So this is a preliminary data showing the relation between CD200 cutaneous form and cutaneous diffuse form. And another experiment that we try to see if how works CD200, and also the receptor of CD200, also important for that infection. Acknowledgement for my lab collaborators in Brazil and also in USA, that is Norma, and also the founding that is FAPESP, that we got the money to do all this research. Thank you very much.
Thank you very much for a very clear presentation. I'm sorry we didn't have a chance to finish off the things there. Just time for one very quick question, please, if anybody has one. Okay, if not, we'll move on to our next speaker.
And now,
we will invite Dr. Sheikh Salam from the Amar Singh College, India.
Thank you so much, ladies and gentlemen. I'm Sheikh Tanveer Salam from J&K, India. So I will be today talking about the prevalence and the pathology of Heterox gallinarum in free-roaming domestic chicken of Kashmir Valley. In fact, this is a part of my study, 2-year study, PhD program. So the context of the research is, since these free-roaming chicken, since they are freely roaming here and there, so they are very much exposed to the germs. So there are a lot of chances of getting the infections. So, and in this study, we studied in fact the Heterox gallinarum infection, which is usually subclinical, and Heterox is considered to be the harmless nematode in the chicken. But there is potential that this Heterox, it acts as a vector for another very nice parasite, that's a protozoan parasite, that is Histomonas. So the present study was designed to have idea about the prevalence of the nematode Heterox gallinarum in free-ranging chicken and to study the extent of pathology caused by it, either during the monoinfection or the co-infection with the Histomonas protozoan. So, this Heterox gallinarum, this shows the life cycle of the Heterox gallinarum and the Histomonas, the association between the two parasites. This Heterox gallinarum, you can see there is the chicken. You see the chicken, these Heterox nematodes are in the chicken, then these nematodes inside the chicken, they just engulf the histomonads. Then those histomonads are inside the eggs of the Heterox, and when these eggs come out in the soil, they hatch into the larvae. These larvae can either be infected by the earthworm, or they can be either directly ingested by the other very vulnerable birds like turkeys, and this can cause the death, even mortality in these very vulnerable chicken if the chicken take either the larvae directly or the intermediate host earthworm, getting into the carrier bird, again infecting the ovaries of these cecal worms and remain inside the eggs. So the methodology for research was, we studied in fact the prevalence and the pathology of the nematode. So sample size was to 35 females weighing about 1 to 2.5 kg, collected from different areas of the Kashmir Valley. Then clinical examination was carried out as floated worm eggs were identified as per the thin port. Then fecal egg counts were undertaken within 24 hours of the laying, and this as per the modified McMaster technique with a sensitivity of 50 eggs per gram. Impression smears were taken from the liver, and the scrapings of the ceca and the intestine were stained with Giemsa and Gram stain. Then for pathological studies, routine procedure was followed as per the Bancroft and Gamble. So then McMahon's periodic acid shift base was used, the stain was used to demonstrate the protozoan inclusions in the sections. Then Grocott stain was used to differentiate it from the fungal infections. Then micrographs were taken. So now coming directly to the results, this is the nematode which we collected. In fact, these are the worms, Heterox worms. This shows the photograph, anterior and the posterior view of the Heterox worm. You can see the prominent esophageal bulb. Then these are the eggs of the Heterox, which are more or less more barrel-shaped to differentiate them from the other ascaridid eggs. Then prevalence was found to be, for the first year it was 3.43%. For second year of study, it's a 2-year study in fact. For second year, it was 5.3%, giving overall percentage of 4.39%. The range of intensity of the worms was 2 to 55, and the mean intensity was 20 was mean intensity, plus minus standard deviation. Then fecal egg count was less than 40 eggs per gram of the feces. And only 2 reports were there, 2 cases were there where there was a co-infection of the Heterococcus gallinarum with the histomonas. One was found in the September 2012, another in the August. So giving overall prevalence of the co-infection only 0.42%. So this shows the month-wise occurrence of the Heterococcus gallinarum. You can see here during the warm months, during the August and the September, there was a peak of infection because these months are usually there is monsoon season in India during that time. That's why temperatures are very high and the rainfall is very high. So that's why, because of the high temperatures, the birds have the lesser immunity and there is readily availability of the host. That's the earthworm. That's why the intensity of infection was very high during these months. So then you can see mean intensity also very high during the during the summer and the autumn seasons. You can see the summer and the autumn season showing the very high mean intensity of the infection also. And then regarding the pathology, both the mono-infection, pathology of the mono-infection and the co-infection was studied. During the mono-infection, it was found that the adults were found in the lumen along with the debris, and the larvae were penetrating, it has been reported that larvae could penetrate the epithelium of the cecum. Then there was intense chronic diffuse inflammatory processes were seen with mononuclear and polymorphonuclear cell leukocytes infiltrating. They were extending up to the submucosa. There was mucosal erosion with parasites and cellular debris in the transverse sections, but no gross lesion was found during the mono-infection. You can see here certain sections. You can see here this is the adult nematode section. Within the section, it is in the lumen. Then these are the larvae, which can penetrate the epithelium of this mucosa. So then this shows the toildune blue section showing the multiple sections of the parasites, these Heterococcus gallinarum. So causing the mucosal denudation. And here you can see the infiltration of these mast cells. Then during the co-infection, in fact, histological sections were taken, histological studies of liver and ceca was taken. And during co-infection, liver was found to be brilliant greenish. During co-infection with the histomonas, in fact. So liver was found to be greenish with numerous round ovals. It was circumscribed lesions measuring about 0.5 to 1.5 centimeter in diameter. There was extensive infiltration of the lymphocytes, mononuclear cells, and certain heterophils. And the hepatocytes towards the periphery show the lesions. They contain the punched out oval bodies, which could be stand poised and become very prominent with the PAS stain. Then cecum microscopic examination of the cecal lesions showed the hyperemia ulceration and infiltration of the heterophils, lymphocytes, and the macrophages. Then there was slowing off of the epithelium, and the lumen was packed with the fibrin and the red blood cells and the tissues. You can see here this section. This is the gross. This is the liver showing the greenish coloration. Then you can see these circumscribed oval lesions in the liver, and this is the TS of the liver showing these it's infected with the histomonads, showing the punched out ovular vacuolar bodies, in fact, and they become prominent with the PAS stain. And you can see here the cecum showing the co-infection of the histomonas and the Heterococcus gallinarum. Its lumen is completely plugged with the cellular debris. So simply now the conclusion is that this Heterococcus gallinarum, it's comparatively harmless nematode, but it is having the great potential of acting as a vector for this histomonas. So especially during the warm and the wet seasons, because I told you there is the decrease in the immunity of these birds, hosts, and availability of the intermediate hosts during these seasons. So thus investigations are advisable to know in detail the local immunological response of the chicken intestine to the mono-infection and the co-infection with histomonas, to devise a strategy so that in future we can devise recombinant vaccines and dietary immunomodulations to enhance the gut immunity instead of relying on the antihelmintic drugs, which are posing great threat to the public because they contaminate the meat and the eggs. So to understand we need to understand the extent of the to which this fatal association between the parasites can damage the these backyard chicken, which form the backbone of our poultry industry in India. So, and we also need to study the possible existence of lateral transmission for histomonas without the possible role of the Heterox gallinatum, because during this study we got one sample, one dead host in fact, which was severely infected with the histomonas, and death was possibly because of this histomonas infection, but we could not get any Heterox worm in that host. So there could be possible role of, without this Heterox gallinatum, there could be possibility of the infection, so that need to be assessed. Thank you so much for your patient hearing.
Thanks. Some questions? No?
Thank you.
Okay, our next speaker is Dr. Samar Bashbishi from Mansoura University in Egypt.
However, large-scale administration of this drug alone is of effective drug combination. We thought that as malaria and schistosomes are overlapped in many endemic areas, and since both malaria and schistosoma parasites feed on host hemoglobin, so an effective anti-malarial drug combination would be valuable if we test it against schistosoma. Amongst the anti-malaria combined therapy, which has been also recommended by WHO, is the artemisinin-based combination therapy, which is formed of any of the artemisinin group plus anti-malarial drug. The most recent anti-malarial combination is a compound nifthocane phosphate tablet, which has been recently recommended by WHO for treatment of all forms of malaria, even the malignant malaria, because of high efficacy, over 90% cure, besides good safety, tolerance, and good pharmacokinetic effect. This drug is only single oral dose, which can relieve patient anxiety from repeated administration of multiple drug doses. This drug is formed of artemisinin to nifthocane phosphate in the ratio of 3:1, with the well-known activity of the artemisinin as antimalarial and antisomal, and nifthocane group, which is related to the 4-aminoquinolene, and this group has also documented to have a good effect both antimalarial and antisosoma. In fact, we have already conducted this study, and we published our results in International Journal of Parasitology in 2013, when we tested this compound in vivo against schistosoma mansoni infected mice, and we found marked reduction in worm load, both against adult and larval stage, and also we reported marked decrease in egg counts and amelioration of hepatic pathology. After that, we conducted another in vitro effect using this compound against schistosoma mansoni adult, its 3 larval stages and snail host, and also we document good result, and we published our results in Acta Tropica 2015. All these encouraging results have motivated us to conduct another in vitro study to test if this drug has also a good efficacy against schistosoma hematobium, with the long-term goal is to assess if this drug can be used as broad-spectrum anti-schistosomal drug. In this study, we used the combined artemisinin phosphate, which was provided for us as a gift by Zyman Winward China, and we compare it with praziquantel, which is a positive control drug for schistosoma. The life cycle of schistosoma hematobium is normally maintained at Tudor Bellhar's Research Institute between snail host Polyneus truncatus, shedding of cercaria, and Syrian golden hamsters. We did perfusion of infected hamsters 12 weeks of post-infection, and we only used viable, motile, and intact worm for culture. For in vitro schistosomicidal activity, we used different concentration of the drugs of combined artemisinin niftequine, starting from 1 up to 40 microgram per ml, and we used praziquantel as 10 microgram per ml, and we used negative controls as media alone or media with DEMSO 0.8%, which is the highest concentration of solvent used. Each concentration has been used twice, and we repeat the experiment at least 2 times. We maintain the culture for 72 hours, and every 24 hours we examined the incubated worms for motility, for morphology, and we record any death rate. Then we document our results by scanning electron microscopy for both incubated worms with the artemisinin niftequine compound and the negative control worms. In our study, we document the dose and time response curve of adult schistosoma hematobium exposed to this compound for 72 hours. We found that immediately after incubation with this compound, all worms were contracted, coiled, with loss of matting, but we start to see the mortality rates after 48 hours of incubation at a dose of 20 microgram and 72 hours after incubation at a dose of 8 microgram. With 100% mortality, we are seen at 20 microgram, 48 hours, and 30 microgram, 48 hours post-incubation. Scores, we give 3 as complete mortality, 1.5 as sluggish or immotile but still alive, and 0 completely dead. We found for all worms were normal till the end of experiment. For praziquantel, we record mortality only 48 hours post-infection, and in 24 hours post-infection, the mortality rate was only 45.45. And all worms were contracted but still in matting. But in all worms incubated with the compound niftequine phosphate, no matting at all. All of them were separated, were contracted and coiled. We start to see the 48 hours at concentration of 20 microgram with 100% mortality at 30 microgram. And for 72 hours incubation, we start to see the mortality at 8 microgram and 100% mortality at 20 microgram. For the in vitro effect of this compound on the mortality of male and female, we found that the high mortality rate was seen among female who died more rapid than male. And before this, the incubated female revealed marked change in mortality like convulsion or irregular movement. However, the mortality rate in males were less than females. We document our results by scanning electron microscopy for females. This figure shows the intact female with regular arranged fissures, and here with tegmental spines. From C to F are the incubated females, showing marked edema of the tegmental surface, blebbing, and vesicle formation, which indicate parasite stress. Also, there is marked cracking, fissure formation, and even exposure of subtegmental tissue. For males, this one show normal male with tubercle and covered by spine and regularly arranged fissures. Here, the incubated one showed marked deformity of tubercles and loss of spines. Here, marked erosion and ulceration, crack formation, and marked loss of tubercles and deformity of the tegmental surface. Here also marked edema, and for the gynecophoric canals, we can see marked contraction, irregularity of both sides, and even wrinkling of inner side, which can affect normal mating, we have seen. And also this change in the gynecophoric canal can affect the carriage of female inside male, which is very important for their maturation and subsequent egg deposition. Also, we find marked edema and swelling around the ventral sucker, and in other parasites, marked constriction and deformity, and ulceration in area around suckers. Here, coiled male markedly contracted at the anterior end, and marked deformity of couple of incubated worm. In conclusion, in this study, we assessed the cost-effective anti-malarial parasite, combined artemisinin nifstocin phosphate, on Schistosoma hematobium. We found a lethal effect of this compound on Schistosoma hematobium at 8 microgram per ml. We achieved 100% mortality at 20 microgram after 72 hours post-incubation, with 100% mortality at 30 microgram 48 hours. Marked mortality has been seen among females and males, which is very important because effective schistosomicidal drug targeting female are more potent because it will be associated with decreased eggs, which is the main cause of pathology in the disease. Also, decreased egg load will decrease spread of parasite in surrounding environment. For scanning electron microscope, it revealed marked tegmental alteration, which were more prominent in males than in female, indicating that this drug is mechanism of action is sex dependent, which differ in female than. We have documented that this compound has important potential as novel schistosomicidal drug because our previous result against Schistosoma mansoni and here also against Schistosoma hematobium. We recommend conducting other studies against other schistosoma species like Schistosoma japonicum and snail host. We have also conducted a study against polyester cases, but we didn't publish yet. Conduct other studies to see the possible mechanism of action, comparing the combined therapy in comparison with artemisinin alone and nifstocin phosphate alone. Studies in human with current anti-malarial dose to see if this dose will be effective against schistosoma or we have to adjust the dose. Assess its value to eradicate both parasitic disease and develop the most feasible and cost-effective strategies for the different endemic areas. Thank you.
Thank you very much for that interesting presentation. Do we have any questions, please?
You said this drug is effective for both parasite and snail, and I'm just wondering, is there any side effects to the rats and or if you use in human being, if you use for a long time, and is there any side effects or not?
I didn't check in human. I checked only on mice, but this drug has been already used as anti-malarial in humans. So it's already a marketing drug, but only for malaria.
Okay.
I think we are the first to test it against schistosoma.
So all your experiments are on rats. So not yet used in human being.
No.
Yeah, because the dose may be different. Because it is it can it's very effective to snails, and I think if you use in human being, could be very sensitive. Yeah, very with caution. Yeah.
Yeah, but it's already used against human in malaria. Okay.
Okay, thank you very much.
Thank
we'll
you.
move on to our next speaker.
The next speaker is Dr. Beiyong Shu from the Anyang Medical University in China.
Good afternoon, everybody. Today our body health, the TH1 and TH2 must be balanced. If TH1 or TH2 are not balanced, then we have disease. For example, if our body is infected by Schistosoma japonica, then the TH1 will increase. If our body is infected by Plasma gondii, if our body is infected by Plasma gondii, then the TH1 response is increased. If our body is infected by Schistosoma japonica, then after infection, about 4 or 6 weeks, TH2 response will increase. As we know too, the TH1 will induce M1 polarization or activation. TH2 will induce macrophages to polarization. We are interested in recently that if our body or animal co-infected with Schistosoma japonica and Plasma gondii, then what will happen in body? So today I will introduce our work about the co-infection. This is our precious work. First, we infected mouse with Schistosoma gondii cyst. Then 5 weeks later, we infected some mice with Schistosoma japonica larva. Then after infection, 6 weeks later, some mice died. Then after 9 weeks, we killed the rest mice. This is one group. Another group, we just infect Plasma gondii. Then 6 weeks after infection, we found there is some mice died. Then 9 weeks after infection, we killed the rest mice. This is a normal control group. The results show that the mouse just infected with Schistosoma japonica. The death rate is lower than normal. But if mouse were co-infected with Schistosoma gondii, with Schistosoma japonica and TG, then the death rate will lower than this group. This is one of the results. That means if mice were co-infected with two worms, then the death rate are higher. Why this is happening? Okay, because we suppose if the mouse first infected with Tigandi, then the Th1 response increased. Then if we infected the mouse with another worm, Schistosoma japonica, then the Th2 response are difficult to increase. So the death rate is higher. Then we killed these mouse and see the liver pathology. We found co-infection. The mouse with co-infection, the granuloma is smaller. But infection with just Schistosoma japonica, the grandma is larger. This is a normal control. If the granuloma is smaller, then the toxin from the eggs will released easily and enter the blood, or will come out and damage the liver cells easily. So the co-infection with the mouse with co-infection is more easily died. This is the staining with muscle. We can see the fibrosis of liver. In the co-infection group, we can see the fibrosis. The fibrosis is lower. So the toxin in the eggs will easily release and do some damage to the liver cells, or enter blood and do some damage to our body. So the mice with co-infection will easily die too. I mean, the response, the immunity response, just two-edged sword. On one hand, the immunity response will do some good to our body, to protect our body. But on another hand, if the immunity response is too high, then it will do some damage to our body. So, as researchers, we must understand how to control the immunity system, the immunity response, and let the immunity response do some good to our body. This is the result of the mouse, of the experiment. In the experiment, we can see, besides the damage of the liver, we can see some damage appear in the blood, in the blood serum. It shows the granuloma, although the granuloma and the fibrosis is lower, but TGF-beta 1 and interleukin 4, interleukin 13 decrease too. RAOS increase, this is decreased. And nitrical oxygen increased, infrared gamma increased, and so on. These results suggest Th1 response increases, which draws M1 activation and inhibits Th2 response. So we suppose that M1 perhaps plays an important role in decreasing the size of granuloma and the decrease of fibrosis of liver in schistosoma mice. If M1 is transferred, we suppose if M1 is supposed to liver of mice in a proper time, and in a proper number, they may be prevent or reverse hepatic fibrosis. Then first, how to obtain the persistently activated macrophages. Yeah. Our previous study showed one macrophages were infected by PRU strain of Tigandi in vitro. They could be activated to M1 cells. One infected by WH3 strain, they could be activated to M2 cells. What is the mechanisms of the genotype associated macrophages polarization? Yeah. Some scientists have do some research. The pictures show if type 1 strain and infect macrophages, then rope 16 were released, and they will activate state 3 and state 6. Then these state 3 and state 6 will end nucleus and let the macrophages to M1. But for the GR15, did not activate state 3 or state 6. But in top 2 strains, the rope 16 did not activate state 3 or state 6. But GI15 activate AF-gamma B, and activate the macrophages to M2 cells. So we do our present work. First, we use the PRU and WH3 strain of Tigani. And take the total RNA from the 2 strains. Then back the RNA to cDNA. Then we use the special primer to let cDNA use PCR to produce a plasmid. Then we put the plasmid into a lentiviral vector. Then we use the viral to infect macrophages. Then we found the macrophages become transform M1 or M2. Then we co-culture M1 with HSC, JS1 cell strain, and detect some marker. This is the results. This is the result of PCR. This is the GI15. This is a rope 16. This is a GR15 PGFP plasmid. This is a T plasmid. This is rope 16 T. This is rope 16 PGFP plasmid. Then we infect macrophage with the virus. This is M1. That macrophages to become M1 and M2. This picture shows the transfection efficiency. The efficiency is about 9.3%. This is 70.6%. These results show that in the macrophages, there is GI15, and GI16. These results are showed in M1 cells. We can the GI15 protein drop makes RNOS increase, TNF alpha increase, and TGF beta 1 low increase and low decrease. But interleukin 12 increase. Especially, we found the MMP13 increased. The MMP13 is a enzyme, is a protein enzyme. It can dissolves collagen. Besides, we found the GI15 protein can make the TNF alpha, nitric oxygen, and urine increased in the subretinent of macrophages. These results show the 16 protein drops macrophages to M2. We can see the ARG1, interleukin 10, and the TGF beta 1, interleukin 6 increased in cells. And interleukin 10, TGF beta 1, interleukin 6 increased in subretinent 2. This shows that we use the Western blotting to tag RNOS, MMP3, and RG1, and found in M1 the RNOS and the MMP13 increased, but in in MM2, the RNOS and MMP13 increased, but in the M1, the RG1 increased. Then we concatenate the M1 or M2 with macrophages cell stream. It shows that alpha SMA increased M1 concoach with GS1. GS1 is a kind of HSC cell stream. Besides alpha SMA increased, the collagen 1 increased to 2. It suggests that GS1 activated by M1. and GS1 secret collagen. This is the protein. It shows the protein increase in ML. The alpha SMA and the collagen are increased in co-cultured with GS1. Conclusion. Our work shows one, lentiviral infect macrophages. Then the macrophages, then the lentiviral will enter the nucleus. Then the nucleus will produce GI-15. The GI-15 will activate AF kappa B. Then AF kappa B will produce INOS TNF alpha and interleukin 12 and MP30. It means the GIF-15 activate macrophages to M1. And the ROP16 will activate macrophages to M1 because we can see the IgG1 increased, interleukin 6, interleukin 10, and TGF beta 1 increased too. The M1 co-cultured with GS1. Then M1 will drive GS1 to secrete M2 to co-culture with GS1. Then M2 will draw GS1 to secrete alpha SMA and coach 1. The alpha SMA increase means the GS1 are activating. The coach 1 increase means the HSC secrete some collagen. That means the M2 cells will produce some collagen 1. Will make the liver to fibrosis. But M1 perhaps, but M1 will inhibit liver to fibrosis. That's all. Thank you for your attention. Okay.
Questions, please. Questions.
Good afternoon, everybody, and welcome to the final session. There's a small number of survivors left. I know there's lots of people that are very, very tired and jet-lagged, but thank you for coming to this session. One announcement, sort of housekeeping announcement. If anybody needs to have parking vouchers for cars, to go to the Omics desk afterwards and they'll sort you out. Okay, we'll start the next session.
The next section is Dr. Kiki Ngel from the Federal University of Nufu-Alike, Nigeria.
Which is your presentation?
US presentation.
That one there?
Yeah.
That one,
yeah.
Okay.
So, yeah, Ngele Keke, Dr. My name, a research paper on co-infections, urinary and intestinal schistosomiasis infections among primary school pupils of selected schools in Ogwelgia, Enugu State, Nigeria. Yeah, that's my school and my name. Schistosomiasis, also known as bilharziasis or snow fever, is named after a German theater, Bilharz, the first person to discover the parasite in Egypt. Then it's one of the oldest parasitic diseases, second to none in prevalence among waterborne parasitic diseases. About 700 million people worldwide may be at risk of infection. People are mainly exposed to the infection as a result of their agricultural, domestic, recreational activities with infected water bodies. And most people live in poor communities without access to good water are actually affected with this infection. And infection is of great health, public health importance in Nigeria, even in Nigeria and in Brazil and all that. The infection is caused by 5 different species of flatworms, which include Schistosoma mansoni, Schistosoma japonicum, S. macongi, and these ones cause intestinal schistosomiasis, and others are S. hematobium and S. intercalatum, which cause urinary schistosomiasis. And the intermediate hosts are mainly the Oncomelina, the Neutriclina, and the Bullionu species. And when an individual comes in contact with infected water bodies, as I said, the sick carrier enters through the skin and further matures in the body. The disease infects everybody, mostly children who are always in contact with contaminated water bodies. And the pathological effects include the enlargement of the liver, the spleen, loss of weight, abdominal pain, accompanied by the deposition of eggs in the urinary bladder that cause what you call the hematuria, the blood in the urine. The research work was carried out in Ogwelgia. Ogwelgia is in Enugu State, Nigeria, and primary school pupils were selected which studied in those areas, and there are a lot of water bodies and rice paddies around the area. The intermediate host of the schistosomiasis, the snails, had already been reported in that area by those people. And the aim of the study was to assess the prevalence of both the urinary and intestinal schistosomiasis among the primary school pupils in that area. Now, the materials and method. The study was carried out, as said, in Ogoelgie in Enugu, and the area is dominated largely by Igbo-speaking people. The local government area has the following communities: Sabo, Gugu, Ihe, Newe, Obaku, Itukuozala, Ogugu, and all that. And there are many primary schools in the area, which include our local government area, which include the community primary school, Agbogugu, Central Primary School, Ihe, Obako, and others. Now, the area covers about 8.35 square kilometers, having over 95,860 persons. And there is electricity and no adequate pipe-borne water in the area. And so that's why the people are always involved with the streams where they go and bath and then do their laundry activities. As they go there, they are always reinfecting themselves. And as they drop their urine and feces or the stool inside the water bodies. Then the rainfall pattern in that area is just from April to October, while dry season is from November and March. Crops grown include rice, cassava, maize, vegetables, and all that. And methodology, there was a pre-visit to that area because the place is made of a mix-up of religious activities and people, the teachers, the village head, and the parents. They were communicated and visited and talked to about this research, and they all bought the idea, and then people were recruited, and they voluntarily responded. There was no compulsion on that. Now, sample collections, 620 sterile bottles for collection of urine and stool were distributed, and then 310 pupils from 5 different primary schools within the local government area, which were chosen at random, were recruited for that. And then each of them were given 2 sterile sample bottles, one for urine and then one for stool sample. And those are the primary schools that were included, that were used for that: the Abogugu, Kebi, Obako, Owelikot, and all that. Now, about 20 grams of stool samples and about 10 to 15 ml of urine samples were collected between 10 a.m. and 3 p.m. when the excretion of schistosoma X were high, and were preserved by adding 2 drops of jik, domestic jik, that's the bleach, and then on the urine, and then while the stool was preserved with a formalin, the formaldehyde solution, 1 ml, and then the research was carried out between November 2012 and October 2013. Now, the examination of eggs, as described by Chesbrook, and we use the examination of urines for the X of schistosoma. That's all the samples as we took them to the laboratory for parasitological laboratory at that institution, Federal Polywana. Then about 10 ml of the urine sample was centrifuged at 500 RPM for 5 minutes. The supernatant was discarded and the sediment was transferred to a slide. It was then covered with a cover slip and examined microscopically with 10 × 10 objective for the number of X, and then was counted and identified and recorded. And then combined test strips were used in testing for the presence of blood. That's the urine, those that have urine, and then the combinant was manufactured by that company in Germany. Examination of stool samples for the X of schistosoma, the method described by Chesbrough and Rara, were used for the examination, and about 10 ml of formal detergent solution in a universal container mixed with about 300 milligrams of stool sample was used. The whole preparation was viewed using times 10 objective, the number of eggs in the specimen were counted, identified, and multiplied by 3 to give an approximate number of gram in physics. Now, the result. In table 1, that shows the prevalence of urinary schistosomiasis among the primary school pupils, as you may have that. Then if you look at the table, it's covered at, there's a touch, the pointer, okay? The middle?
Yeah.
Okay, yeah, sorry. Now it's covered at the Akebi Primary School had the highest prevalence, and the total prevalence was 44. And that's the prevalence of pupils with hematuria using the Combi-9. And then Akebi Primary School as well were more infected, which children of that school had blood more in their urine as a result of the schistosoma hematobium, and that's caused that. And the total prevalence was 58.9. Now, children between 12 to 14 years of age, when we were trying to compare the age relationship, were found to be more infected with urinary schistosomiasis. And these children were actually found mainly around the water bodies, playing around with it. They're going to fetch or to swim and all that. And that was the total prevalence of the infection with the age of children. Now, with the prevalence using intestinal schistosomiasis, then children within the same age range, 12 to 14 years, were still more infected because of their constant contacts with the water bodies and all that. Now, prevalence of intestinal schistosomiasis among primary school pupils in Ogo is that with respect to primary schools, then this primary school, the Akebi, was more infected with intestinal schistosomiasis because the school there is always surrounded. It's surrounded with water bodies, and then the rice paddies, a lot of rice paddies around there, and children are always moving around there to wash their clothes, to swim, to do all the recreational activities. And that is the total prevalence among that. Now, that is the prevalence of mixed infections among the intestinal primary schools in Ogo. And in the mixed infection, we discovered that Akebi Primary School were the most infected, both with the intestinal schistosomiasis and then the urinary schistosomiasis. Because the school there, as we said earlier on, there are a lot of water bodies and a lot of rice paddies. So the children from the school, they're always around the water bodies, just as they are there, they will be playing, urinating right inside the water bodies, and then passing their defecating right inside the water bodies too, with their stool and their urine, and then there will be reinfection among them. And that is the total prevalence of the infection in that area. Now, just to say the discussion, it was actually found out that the children, primary school people in that area were mainly affected with the intestinal and urinary schistosomiasis, just as a result of their constant contact with water bodies. And that's the prevalence, as we've already seen. then with a urinary and intestinal schistosomiasis, and respectively from Akebi Primary School. And those are the, even we discovered that the children who always, children were always in contact with the water bodies, which they use for recreational activities, swimming and fetching for domestic purposes. And that's really increased the activities and they equally increase the infection of the of that parasite on the children. And the prevalence of infection, was this 58% among the pupils tested for hematuria using the Combi9, and which was very high, to show that that area was highly prevalent with the infection. And those are the people that already worked on those that had a similar result on that kind of work. Like you said, the test strip revealed high endemicity of level of urinary schistosomiasis in the area, just as reported by others. And children between 12 to 14 years were the most infected with urinary and intestinal schistosomiasis, and 48 and 15 respectively, which when it was tested, it was be less than 0.05 than other children sampled during the study. So theirs was much higher. And then I would say children of this other age bracket, 14 to years, were very active playing with the water bodies. That they were more prone to infection of the schistosomiasis, both intestinal and urinary. It's almost something that we have. Then the prevalence of co-infection, as we had, is 41.61%, which was equally high, and which equally was reported in other places by those researchers. And then the high prevalence of co-infections of urinary and intestinal schistosomiasis makes the area endemic for both urinary and intestinal schistosomiasis. So the high result of prevalence of the co-infection is also as a result of high level of water contact activities with the pupils in the area under study. The water bodies and the rice paddies were incriminated with the fresh water snails, the intermediate host of schistosomiasis, as earlier reported by those authors. Now, in conclusion, the results obtained from the research work have referred to establish the facts that the prevalence rate of both urinary and intestinal schistosomiasis co-infections in the study area were 44.84% and 41.61%, which is high, and respectively with the prevalence. With the result obtained, the area can be said to be endemic with both urinary and intestinal schistosomiasis, and water contact activities contributed to the high endemicity level. The health education, as we recommended among the populace on the method of transmission and prevention of disease, is highly recommended, and also provision of adequate pipe-borne water is necessary in the area, and the infected people should be treated. And those are the references. So thank you very much for your listening.
We are open to questions. Okay.
Yep.
Yeah, I realize in
in Nigeria,
Yes.
there's such high prevalence and intensity. So I'm wondering, yeah, for those kids in the school, and do they know they have they will take risk if they swim in the water? For example, for farmers, they have to they work in the paddy field and they get infections. They have to work. They have to work for that they have to. But for the kids, and do they know they will take risk of getting infection of the disease, and do they scale? Do the people scale for the schistoma infections?
Now,
So that's
okay, that's your question.
Yes.
Okay.
My question is to the people,
Okay,
to the
to
kids.
the people now, the advice is that, yeah, there are farmers using the rice paddies, which are always incriminated, to go to the rice paddies with their boots, red boots, and because they cannot do without the rice farming, which should be a livelihood to their families. And then for the children, we recommended that if government can help to sink boreholes, what you call borehole, motorized boreholes over there, that will help to reduce their contacts with the water bodies. And even as they go there they might not be going there for the swimming and then all that. And also, we recommended for the using molluscicide on the water bodies so as to reduce the intermediate hosts of schistosoma, which is always involved in transmitting the infection. Yeah.
I think that's a message, but I think the attitude of the people is very important. You know, in China, when the people know the danger, when they know the danger of getting infections, and the people they scale, they don't want to. Yeah, they don't want to because they fear such kind of activities very much. So I think that's a very important way. Yeah. That's why in China there are many kinds of such kind of parasitic disease was eradicated because
actually we recommended for treatment too. Treatment, if treatment, using praziquantel and vermectin, because work has been done which shows that a combination of praziquantel and vermectin will help to reduce the parasite load and the affected people.
Okay, thank you.
You're welcome.
Another question there.
Yeah.
Thank you. I don't have a question. I just want to add one or two things to the questions you asked.
Yeah.
We have done some work on schistosomiasis in some other parts of Nigeria. His was in Enugu, mine was in Osun State. These children, aged between 12 and 15, whether you tell them there is a crocodile in that river, they will go and swim. They are not afraid of any infections. In fact, some of them think that for them to have blood in their urine is a sign of
maturity.
Their maturity.
Early maturity.
So there is none of them is afraid to go and swim in a river where they know they will catch any kind of infection. They know that they will get the infection, they will be treated, but that fun of swimming in the river, nobody can take away from them. They will still swim.
Okay. No more questions? Only a point. In Brazil, we had some endemic regions in the past, but the combination of health education and prophylactic measures, we reduced a little the infection.
It's okay. All right. Thanks so much.
Okay, our next speaker is Dr. Owolabi from the Ahmadu Bello University in Nigeria.
The first one.
Oh, yeah.
Good evening, everybody. I'm presenting on seasonal infestation of small ruminants by nasal birds in Kaduna State, northwestern Nigeria. Nasal birds are also known as estrus ovis. They are obligatory myiasis-causing flies that infest small ruminants comprising of sheep and goats. They infest the nasal cavities of these small ruminants, causing them to sneeze, cough, and producing mucopurulent nasal discharges. And also, these animals become unthrifty, and it brings about production losses. And most of the times, we have low fertility. And because livestock is an important component of Nigeria's agricultural economy, especially small ruminant production, in meeting animal protein requirements and providing vital raw materials for the agro-based industries, it also provides employment for many Nigerians who engage in production and marketing of livestock and its byproducts. So, information on the prevalence of this disease in each distinct climatic and sociological situation is a prerequisite for the rational design of economical and effective preventive control program. And as such, this has brought about the background of which this prevalence study was conducted. And it was conducted over a period of 24 months, comprising of 2 dry and 2 wet seasons. This is a life cycle of estrus ovis. The adult female fly flicks the larva, the first stage larva, into the nostrils of the small ruminants, where it molts from the L1, L2 onto the L3, which eventually pupates in the soil and eventually becomes an adult. The study was carried out in 5 local government areas of Kaduna State, located in the Northern Guinea Savannah Vegetational Zone, which included Sabongari, Zaria, Giwa, Kudan, Makarfi local government areas, respectively. And the study was conducted over a period of 2 years, from November 2005 to October 2007, covering 2 dry and 2 wet seasons. The heads of these slaughtered ruminants were purchased from butchers in marketplaces and abattoirs on weekly basis from selected local government areas. They were transported in polythene bags to the entomology laboratory of the Department of Veterinary Parasitology and Entomology, Faculty of Veterinary Medicine, ABU, where they were further processed. The heads were hands-on vertically through the nasal cavities, using forceps and pen knives to expose the nasal cavities, turbinate bones, and frontal sinuses. With the aid of a hand lens and a pair of hand forceps, larvae were collected and examined in situ. That's the larva in situ, the nasal cavity, and the turbinates. Sections of the nasal cavity and turbinates were processed using the technique H&E, and histopathological lesions using the method described by Kiernan. Statistical analysis were carried out, chi-square used to test for level of significance between the wet and the dry season, and ANOVA was used to test prevalences between the different local government areas. The results, we could see that Sabongari had the highest prevalence for infestation, while Kudan had the least infestation. And during the dry and the wet season between the sheep and goats, we could also see that there was a high level of prevalence during the wet season, which was actually what entails, because during the wet season, though for the sheep infestation, all year round, there was no statistical difference between the wet and the dry season for the sheep. But we could see that for the goat infestation, there was actually a higher prevalence during the wet season. And it might be that Estrus ovis actually prefers the sheep. The sheep is a preferred host for Estrus ovis infestation. So that might be why there was an all year through infestation for the sheep. This is just trying to show us the various prevalence rates in the different local government areas in which I said that the Sabongari local government had a higher prevalence. This could be due to higher activity, higher influx of the small ruminants for slaughters and all that. And here is still showing us that we have, usually between the months of April, May, June, there's a spike in prevalences for the months, which coincides with the rainy season for the first year. And even in the second year, we can still see that April, May, June, and August, which happens to be the rainy season equally. So there are spikes of prevalence increases during these months. And we can see, this is a photomicrograph of the turbinate tissue from sheep of non-infested. We can see that it's all normal, there's no congestion. This is a photomicrograph of the turbinate tissue, you see. It's all normal, there's no congestion. While in the infested, you see the venous is highly congested. In the submucosa, you see congestion all around due to the infection. So a lot of mononucleosides and all that's all invasive. You can see all congestions in the turbinate tissue. In conclusion, prevalence of Estrus ovis in small ruminant head studied is an overall of 20%. It is significantly higher in sheep than in goats. It is significant difference in the prevalence of, there is significant difference in the prevalence of Estrus ovis in sheep and goats in all the 5 local government area studied, even though they are located in the same vegetational zones. Although in humid and tropical countries, adult fly activity and larval development occur all year round, Estrus ovis infestation was observed to be significantly higher during the wet than dry season in goats, but in sheep, there was no significant difference in rate of infestation in the dry season compared to the wet season. Histopathological studies of the infected sheep and goats showed congestion of the venous sinus in the submucosa, whereas the uninfected sheep and goats showed no histopathological lesions. Thank you all for listening.
Thank you for a very clear presentation. Are there any questions, please?
Is there any human infection with these flies in the local areas?
Can't get you.
Is there any human infection with these flies in the local areas?
Yes, there is. Yes. You have sometimes when you have high activity, human activity around these areas of flies, sometimes humans get infected. You have ophthalmic myiasis, which could be taken out manually, by manual traction. Yes.
What's the clinical manifestations of
In the ruminants or in humans? In humans, usually you have conjunctivitis, watering of the eye, pain, inflammation around the eye area.
Sinus?
No, not necessarily sinus. Not necessarily sinus.
In the same way, and which is the mortality in humans for this?
No mortalities.
No mortality.
Do you get a higher prevalence in humans if there's a high prevalence in the sheep and goats around?
That's if the sheep and goats are not readily available, then they go for the humans. But once in a while, there's accidental infestation in humans. Yes.
Okay, thank you very much.
The next speaker is Dr. Geoffrey Hyde from the University of Salford, United Kingdom.
Good afternoon. What I want to talk about this afternoon is some work that we were doing, developing a technique called mobile genetic element PCR as a tool for investigating the spread and infection of parasitic diseases. So what I want to talk about in the session is to tell you a little bit about what MGE PCR is, and also to give you perhaps a couple of examples of how we can use it. First of all, the spread of human sleeping sickness in Uganda, and also strain typing in Toxoplasma. So there are a wide variety of markers, genetic markers for identifying parasites. So examples are the rapid markers, restriction fragment length polymorphisms, microsatellites, and each of these marker systems have their own specific uses and specific advantages. Sometimes the limitations of these techniques is that they require multiple stages. So for example, multi-locus restriction fragment polymorphism requires perhaps several different stages to get the results, or the combination of different data sets. So what I was interested in doing was trying to produce a method that would give you a very simple single stage approach to discriminate between parasite strains. So the principles of MGE PCR, so MGE stands for mobile genetic elements. So I'm sure many of you are aware that in the genomes of many species, including parasites, including protozoan parasites, you have mobile genetic elements, pieces of DNA that move around between within the genome. And the principles of this technique are that in any two individual strains, or any multiple individual strains of parasites, there will be differences in the position and perhaps number of these mobile genetic elements. And if we can develop a mechanism of identifying the positional variation in these elements, it gives us an opportunity to look at differences between those strains. So for example, in this diagram here, the top strain has 3 mobile genetic elements. The second strain, strain B, has lost one of those mobile genetic elements. And if we can detect these differences, it gives us an approach to looking at strain variation. And particularly if we can develop PCR primers that recognize those mobile genetic elements, we should be able to amplify fragments from them. So for example, in this simple diagram here, if we've got PCR primers between the elements like that, we'll get a different banding pattern on a agarose gel compared to between the two strains. So typically the structure of a mobile genetic element is an element of DNA. Very often at the two ends of the mobile genetic element, they've very often got an inverted repeated sequence of DNA. And that offers us the opportunity to develop a single PCR primer that recognizes both of those inverted repeats. So if we design those primers effectively pointing outwards from the mobile genetic element, what they will do is they'll amplify the DNA between adjacent mobile genetic elements. So we need to know nothing about the DNA between the mobile genetic elements, just that there are these elements present. So this is the basic principle of the technique. So I want to give you some examples now of how it can be used. And the first example is looking at the origins of human sleeping sickness epidemics in Africa. And this is some work I've done in collaboration with a group at the University of Edinburgh, where we've been looking at in detail a couple of sleeping sickness epidemics in Uganda. So the particular questions here are around about the sort of location of the epidemics. So there tends to be human sleeping sickness in Africa tends to be restricted to fairly localized foci. And so the question is really, why are these foci so defined? And then what causes these occasional epidemics to occur? So this gives you a bit of an idea of what I mean. So this is a map of East Africa. The red areas are kind of historical foci of human sleeping sickness. And as you probably know, human sleeping sickness and animal trypanosomiasis is transmitted by the tsetse fly. And you find tsetse flies across this whole region. You find animal trypanosomiasis across the whole region. But human disease is localized into these foci that I've indicated in red on the map here. Also, when you track the number of cases in certain areas, either locally or nationally, this is the pattern of disease in Uganda, and you see this kind of fluctuation in numbers. Some of these differences in prevalence are caused by socio-political reasons, social unrest, etc. But if you look at local areas, you find that sometimes you get the flare-up of epidemics. So we've got these particular issues. We've got the fact that there's animal disease throughout the area. Human disease seems to be very localized, and then this human disease occurs in epidemics. So my interest really is in what causes this kind of pattern of disease. So if we look specifically in Uganda, what we see at the moment is that there's a spread of human sleeping sickness from a particular focus in the north up to the northwest. So the area I'm interested in is at the north end of Lake Victoria, which is this large blue area here. So this is the focus that we're interested in here, the Busoga focus. And this has been a historical focus for a very long time, but currently what's happening is that the disease is spreading and it's moving northwest, and it has the potential to merge with, if you remember, sleeping sickness has an East African form, TB rhodesiense, and a West African form, TB gambiense. And the only place in Africa where the two diseases are really close by are in this area here. And this particular movement of disease is approaching an area which has the potential for causing great difficulties. So I want to just discuss a couple of epidemics which we, in collaboration with my colleagues in the University of Edinburgh, have looked at in detail. An epidemic between 1988 and 1992 in an area called Tororo, and then a more recent epidemic about 10 years later in Soroti in Uganda. So these are the areas here. So Tororo, just northeast of Lake Victoria there, and Soroti to the north of the map there. So the first epidemic I'm talking about is in Tororo. So in this particular epidemic, there was never any sleeping sickness prior to 1984. The epidemic peaked around 1990, and it's still grumbling along. There's still a few cases of disease there. We did a lot of work, and I don't have time to talk about the details of this in relation to this talk, but we did a lot of work on that epidemic. And what we showed was that the epidemic was defined by a single human strain or a strain group, which seemed to be spreading amongst the people there. It was mediated by cattle, so we did some epidemiological studies that showed that transmission of the parasite from tsetse fly via cattle to tsetse fly was 5 times more probable than it was going from human to human. So in other words, what this showed was that cattle were a significant reservoir for human sleeping sickness. So we identified the presence of a specific strain, which we think is what defines these foci, and that the cattle are an important reservoir. So moving on to this more latter epidemic. So again, never been any reported cases of sleeping sickness. On New Year's Day, New Year's Eve of 1998, a local news nurse reported the first case. And subsequent to that, there were a large number of cases over the next few years. And one of the characteristics of this area was there was the importation of cattle as part of a government restocking program. So there was the suggestion that maybe what was happening was that cattle were being moved from areas like Truro with the presence of sleeping sickness, with the presence of these human infective strains, and the cattle were being moved from Truro to places like Soroti, and that the disease, and then the local tsetse flies in Soroti were biting the cattle and then passing the parasite infection on to the human population. So there was this, an association with the local cattle markets. And my colleagues in Edinburgh did a study where they looked at the distribution of cases both in relation to time and distance from the cattle market. And what they showed was that this cattle market here was the epicenter of this infection. So the evidence was stacking up to suggest that the cattle moving between areas like Soroti with sleeping sickness into this area were responsible for the importation of the disease and therefore potentially the stimulating the epidemic. So the question then was really where did the parasites come from? Was there any way of tracking the parasites and trying to identify their origins? And so we needed a method to accurately identify the strains, and this is where we utilised the MGE PCR as a tool to do this. So in trypanosomes, there are a number of mobile genetic elements, but this is one of them. It's called the rime element, and it sometimes has an insert called the INGI insert. But essentially, it has the characteristics that I showed you before of a mobile genetic element with these inverted repeats at the end of the element. So that provided the opportunity to develop the MGE PCR in this system. So effectively, what we're doing is analysing the position of rime elements by PCR amplifying between adjacent mobile genetic elements. So this is the kind of pattern that you get. So there are large numbers of these mobile genetic elements present in the trypanosome genome. So effectively, each lane in the gel here is an individual isolate, and effectively what you get is a molecular fingerprint of each isolate. And this is basically a single PCR which you run on a gel, and these are the kind of patterns you get. And as you can see, there's different patterns in different isolates there. So this question was then, if we use this technique, can we identify any of the patterns that were present in Truro and the earlier epidemic in relation to the more recent epidemic in Soroti? So this is, you analyze the data by generating a dendrogram, and if we look at the data, we can see that if we look at some of the strains that were isolated from the epidemic in Truro, we can see that there's the human infective forms, so we call these TB rhodesienses, the human infective trypanosomes, and the animal infective or the non-human infective ones are located in a different genetic group. And when we look at the strains from Soroti, from the humans in Soroti, we find that they come from two sources. So there are some that are located within the Truro group, so they have genetically identical or very similar fingerprints, molecular fingerprints to the ones from Truro, but we also find this group here that are different, and they have a different set of fingerprints. So what that suggests is that some of the human parasites as taking part in the epidemic in Soroti have been derived from Truro, but others have come from another source. So our kind of overall conclusions is, yes, there has been some movement of cattle from Tororo to Seroti, taking with them human infective trypanosomes, but also there must be sources from elsewhere. So we need to identify those more clearly. So that's kind of an example of how we can use these mobile genetic elements to investigate a question about the origins of parasites in this example. And clearly one of the things we've identified is that the origins of this epidemic are rather more complex than we originally imagined. Cattle are important, and that maybe we need to identify specific risk areas to maybe inform people about where they should be moving cattle from and to, and where they should restrict that movement. Just another very quick example to finish off with. We've also developed the technique to strain typing in Toxoplasma. Professor Shen has spent a lot of time this morning talking about the different Toxoplasma strains, so I won't elaborate much more than that. But if we look at just some of the classical strains, type 1 strains are often associated with virulence to mice, and type 2, type 3 with avirulence. And we can use the same technique, mobile genetic element, PCR, to develop a molecular fingerprint for strains of Toxoplasma. And here again, you can see there are different patterns there. And when we generate a dendrogram from those strains, we can see a very clear distinction between the virulent type 1 strains that are down here. These are just reference strains, and you can see these are all type 2 strains up here. But what you can see is that within the type 2 strains, there is a degree of variation, and that probably reflects the rather larger number of multilocus genotypes that Professor Shen was talking about this morning. Okay, so hopefully I've given you an indication of what mobile genetic element PCR is and how it can be used perhaps as a tool for tracking and understanding parasite epidemiology. Thank you very much.
Nice talk, Doctor. I have a question. Do you think that it's possible to do the same with Leishman-Chagas? Because I had a question to this morning. The Leishmaniasis in São Paulo state, it's supposed that that is not from Northeast Brazil, but from Bolivia.
Yeah.
Is it possible?
It should be. I can't remember. There will be, I'm sure, mobile genetic elements within the Leishmania genome, so it will be a question of identifying a suitable Leishmania mobile genetic element, developing a primer to those inverted repeats, and trying it out. I think it's probably would be a very good tool for that question that you've just suggested here.
Questions? Hold a minute, please.
Sorry, yeah.
We use this tool to track the malaria in the border.
In principle, it should work on a wide range of parasites. So we've tested it on Toxoplasma and on trypanosomes. It's been used in Clonorchis, the trematode. So in principle, it should be as long as you can identify the sequence of mobile genetic elements. It should be a generic tool for a wide range of parasites. Yeah.
I was wondering, you're talking about moving cattle elsewhere to occur another thing. How about the wildlife in Africa? Is that also a role to spread the disease elsewhere?
Yeah, I'm sure it is. I think one of the well, to be very frank, there's very little work being done on wildlife simply because collecting the samples is a challenging thing, and to collect and analyse the parasites is challenging. The group in Edinburgh have done some work in the Serengeti where they've been looking at wildlife, some of the larger ungulates and things as potential carriers, and they're certainly infected, but I don't think we've done any strain typing to determine whether they're human infective ones or just animal infective strains. Yeah.
My question is, what do you think is the possibility that in the future could be used other mobile elements, for example, microRNA, not coding RNA, for detect parasite strains?
Yeah, microRNA should be the key thing. I think is to have these repeated elements. If they've got repeated elements, then yes, that should be possible. Yeah.
Okay. Thank you.
Hi, Dr. Jeffrey. And I'm just wondering, you see, is the mobile is a kind of a mobile element? Is that very convenient to bring some instrument with the people when they do the investigation? Is it or just a kind of technical?
Sorry, could you repeat?
Which that means is a specific primer or the technical? Which is more important?
Oh, for any particular parasite, you need to develop a specific primer. So you need to have some information about the mobile genetic element initially, but you don't need to know anything about the position of the mobile genetic element. Is that
so? Yeah. It is only for this parasite, and you find some very specific region or primers. You can divide. It's virulent or avirulent. But for other species of parasite, you may not. It would be it's possible. It may not possible to find a very specific region for segments for such kind of division.
Yeah, so I think you would need some genome data for a parasite with no genome data. It would be very hard to develop this kind of approach initially.
And that means the development of the primer is more important.
Yeah.
And thank you.
Okay, thank you. Okay, our next speaker is Abimbola Amu from the Olabisi Onabanjo University in Nigeria. Do you know which is yours?
Parasit 2015. I can see it. Let me put on my glasses.
Sorry, did you load it up earlier?
Yes, I brought it in the morning.
Yeah.
I brought it and the guy.
Yeah. It's not that one. Not that one there, no.
No, that's not the one. No, presentation.
Okay, have you got it on a stick?
Okay, let me go and bring it. Get my flash drive.
Okay. If you could just bear with us for a minute or two, please. I wonder if it's that one there. No. Not that one. Is that it? Okay. Okay, great, good.
Good afternoon. I'm presenting a short talk on Beyond Fighting Malaria with Drugs and Treated Nets. And this talk is as regards with my country, Nigeria. Advocacy for total integrated vector management. As we know, malaria is an infectious disease caused by the protozoan parasites of the genus Plasmodium and is transmitted to man by female Anopheles mosquito. The etiology varies, and there are about 5 species of Plasmodium: Falciparum ovale vivax, malaria, and Knowlesi. Knowlesi is found in Southeast Asia. It's not yet in Africa. And more than 95% of the species that happens or that causes malaria is Plasmodium falciparum in Nigeria. What is the burden of this disease? In Africa, 45 countries are endemic for malaria, with a total population of 588 million people at risk. Nigeria accounts for 25% of all cases in Africa. The burden in Nigeria is enormous. 97% of the more than 150 million people are at risk of malaria. 60% of outpatients' visits to the hospital is due to malaria. 30% of hospital admission is due to malaria. 11% of maternal deaths is due to malaria. And about 300,000 childhood mortality is due to malaria every year, with a total financial burden of about 132 billion naira, which is close to $1 billion. The national response. If you go down memory lane, the pre and post independence, there actually was a mosquito destruction law enacted in the southwestern part of Nigeria. The regions at that time had some autonomy, so each region had to enact its own laws. In the southwestern part of Nigeria, we had a mosquito destruction law. And this law gave birth to the employment of sanitary inspectors, whose duty was environmental cleanliness, and they adopted the drain and cover policy. It means they were empowered to go into households, to look into the cleanliness generally, and advise and even prosecute people who left water uncovered or who had dirty environments where water could aggregate, which will help mosquito breeding. But after independence, this thing went away. Control at that time, after the advent of the inspectors, was an overall dependence on case management. And quinine, which has been banned for malaria, was the drug of choice. There was also some herbal self-medication and the advent of drug resistance to quinine. In the last 15 years, the country has had 4 strategic plans: 2001 to 2005, 2006 to 2010, another one 2009 to 2013. All these strategic plans were directed towards malaria control. And what were the 4 basic attributes of these plans? 1 was the use of insecticide-treated nets. 2 was intermittent preventive malaria in pregnant women, especially during the 1st and 2nd trimesters. They were given some drugs to prevent the development of malaria. The 3rd was indoor residual spraying. And the last was case management with artemisinin combination therapies. The last strategic plan is the current one we are in, which is 2014 to 2020. And the aim of that one is elimination of malaria by several means. 1, strengthening diagnosis, the use of rapid diagnostic test kits versus microscopy, and that 80% of the population must use preventive measures. Governance has to be strengthened, And all health facilities are supposed to report and notify on all malaria cases so as to be able to, appraise the progress of interventions. My question is this: will these interventions, diffuse as they are, will it ensure elimination of malaria in Nigeria? Of course, the answer is no, for several reasons. One of which is the objectives are too diffuse, with no specific focus. Now, let us look at all those attributes one by one. Indoor residual spraying is supposed to be available to everybody around the country. Only 2% of the population have ever used anything indoor residual spraying. The use of ITNs, yes, it was popular at its inception, but uptake as at 2008 was only 11%. And as at last year, 2014, the uptake has increased a little bit to about 30%. What of the other 70%? How can we depend on these kinds of interventions which do not have far-reaching effects on the population? Intermittent preventive treatment for pregnant women is very, very effective, especially in pre-gravidity. Those mothers with 2, 3, 4 children, a lot of them don't go to hospitals to be given these drugs to prevent malaria. Because one, they have previous experience, two, they can treat themselves. The use of artemisinin combination therapy is still the use and the drug of choice in the country. And there is no resistance to that drug yet. But I know for sure that in this conference, somebody is going to report on imported malaria from Nigeria, on a patient who brought malaria from Nigeria to China and who is not responding to treatment from artemisinin. As far as I'm concerned, in the country, no resistance has been reported for ACT. Now, to contemplate complete control and elimination of malaria in Nigeria, we have to do nothing but kill and destroy mosquitoes. If you look at the epidemiology of the disease, there are 3 important things before malaria is established: man, the parasites, and mosquitoes. Mosquitoes are the weakest link of the triangle. You cannot kill a man for having malaria. You have to treat him. The parasite you cannot see with your naked eye. What do? If you kill mosquitoes, you are reducing the man vector contact, thereby reducing the prevalence and thereby reducing the burden of the disease. I started advocating for this since 2009 because of the privilege of having worked in Florida at a mosquito control agency for a whole year in 2006, 2007. And I was thrilled. I took that option. I had 2 options: one, to lecture in the university for my sabbatical. The other one was to work in the mosquito control agency. And I took that option because I had never worked with mosquitoes before then. And what do? The model, the integrated vector management I am proposing, is the use of the state of Florida as model. If you've been to Florida, it's a tropical state. It's a tropical environment just like Nigeria. It is hot, it is humid, round the year. And Florida has the largest coastline in America. The west, the east, and the southern parts of the state are covered with water. So they have a lot of marshlands, and thus there are plenty and plenty of mosquitoes. Florida has 67 counties, and 64 of these counties have local mosquito control agencies. Why? Because Florida State is a tourist state. The economy depends on tourism. And if tourists come and they are inundated with mosquitoes, definitely they will not come another time. So it behoves on them to make sure to protect all residents or all people in the state against mosquitoes and mosquito-borne diseases. That is why each of the counties, or 64 out of 67, have mosquito control agencies. And the aim is to control, the aim of mosquito control is to prevent residents from mosquitoes and mosquito-borne diseases. And how do they do that? There's a lot of surveillance. They survey for mosquito population, they survey for the environment, they survey for arboviruses from mosquitoes. Also, they do source reduction, which is nothing but larviciding and preventing puddles of water to gathering. They also adulticide. Larviciding is killing larvae. Adulticiding is killing all adult mosquitoes. Incidentally, it is adult mosquitoes that transmit diseases, but you can limit the number of adult mosquitoes when you previously larvicide. They also do a lot of applied research. They have to do applied research so that they will know when to improve already used intervention based on results of research. And they do customer service and do public education. And results of all the above activities are used to make critical decisions on interventions. How do we adapt this to Nigeria? One, there has to be a good political will. Government in Nigeria is in 3 tiers: the federal government, the state government, and the local government areas. The local government is the nearest to the people, and therefore, this is the local government that is easier to use. When you have an aggregate of all chairmen of the 774 local governments in Nigeria at a meeting like this, and you convince them that for them and their children, it is better to kill mosquitoes to prevent malaria, they can come together, agree, based on advice of lawyers, put in a draft law to the legislative houses. And those ones will look into the previous mosquito destruction we had in the 50-57s and merge it together to be operative for the whole country. We will have a law. This thing will not work if there is no interagency cooperation. The hospitals will have to cooperate. All hospitals from the primary to the tertiary will have to cooperate because the law will involve them notifying and reporting on cases of malaria, because it is only the number and the prevalence of malaria as soon as interventions are introduced that you can use to gauge the success or otherwise of the intervention. Are there advantages to using integrated vector management? Yes. The first one is there's going to be significant reduction in vector-man contact, which will reduce malaria prevalence. Second one is that all other mosquito-borne diseases are also controlled, not necessarily malaria alone, not only malaria. Yellow fever is there, filariasis is there. They're all transmitted by mosquitoes. Also, there will be a significant reduction of the disease burden, which we have mentioned earlier. Fourthly, there will be employment. Each local government will employ between 25 to 30 young graduates to do this process properly. Fifth, the people will have a feeling of ownership since they initiated the scheme and are also contributing to its operation. And lastly, energy is concentrated in a focused direction rather than being dissipated on several components with little or no significance to reduction of the disease. There had been so many donor agencies which have been supporting malaria in its supporting the control of malaria in Nigeria. Several of them. WHO, the World Bank, Bill and Melinda Gates Foundation, USAID, the Canadian Red Cross, all have, one way or the other, contributed to buying insecticide-treated nets, which are actually supposed to be distributed to the people free. But when you go to the market now, you find it there, and it costs a lot of money. Some people cannot afford to buy it. They were donor given articles that should actually be given free. But one way or the other, some find themselves in the market and people have to buy. Mosquito control is not an easy thing. It is a job that you do consistently, that you do systematically, that you do conscientiously. Florida started its own mosquito control in 1950. Today, 65 years later, they are very proud that their state is not only free from malaria, it's free from a lot of other arthropod-borne or mosquito-borne diseases. Between 1999 and 2003, there were only 11 cases of malaria in Florida. And it was, those 11 cases came by because there were migrant workers from Southern America who brought the parasites with them. And because they could not afford accommodation in Florida, they camped out in the open, and they were bitten by mosquitoes. The mosquito picked the parasites, and the cycle went on. When you have malaria in Florida and you are taken to the hospital, you are not released until the last parasite is eliminated from your body. You are in quarantine so that you don't become a source of infection to others. In 2009, I wrote an article in the Punch newspaper in Nigeria. It's the wildest, it's the most read paper in Nigeria. And it was at the back page, October 29, 2009. That was the first time I advocated for integrated vector control for malaria. I think the time to start integrated vector management for malaria in Nigeria is now. If we don't start, all those measures which are in current use that we think will eliminate the disease will never eliminate the disease. If you don't destroy the mosquitoes, malaria will always be there. Since we are not sure socioeconomic life of the people will increase in leaps and bounds in the next 5-10 years, I think the time to start is now. Thank you for listening.
Thank you for a very passionate talk there. Do we have any questions, please?
Thank you for your. I think I'm very agree with you. For your aptitude for the vector control is most important for control the malaria. So, and you just mentioned for as I understand the malaria, the mosquito net, it must be important strategy to be used as for the, especially for the children. But you just mentioned for till last year, only that 30% of the person used the mosquito net. I want to know the reason. If it is because of the economic problem, or because some people just doesn't used to use it, or I think it's the malaria case is going down right now. It's controlled, like less and less mortality globally, as the WHO report mentioned. But I think the main point is because people start to use the mosquito net. Why or how to increase the percentage of the people to use the mosquito net?
Thank you. The use of mosquito nets in Nigeria dates back a long time. Shortly after independence, mosquito nets came into the country. Those ones were not insecticide treated. Also, the use of home screens for windows and doors also came in. But the problem with insecticide treated nets is that they generate heat. Nigeria is a tropical country. It is naturally hot. When you now put 2 or 3 children to sleep under a net, which is generating heat, they will not use it. There have been several studies in the country to find out why there had been low uptake of insecticide-treated nets. One of them is heat. The other one is economy. We were not at the hospital when it was distributed, and they have not given us, we cannot afford the money to go and buy it in the market. That is a small percentage. But the majority of those who have the nets and are not using it is simply because it generates heat. Now, the country has introduced long-lasting insecticide-treated nets, which you can use for 3-4 years without retreating. But that still generates some heat. Electricity is epileptic. If you don't have electricity and the fan is not working or your air conditioner is not working, you cannot use that net. So that is the major reason. It generates heat. And some people are allergic to the odor it emits. That is why some don't use it. Or that is why the uptake is low.
Okay, thank you.
Thank you for your interested conference. Thank for your very good conference. I remember that when I was an student, learned that in the human population there are different genotypes for the resistant for the anemia falciparum. Then my question is, actually, there are similar situations. There are different resistance in human population for the malaria?
Well, there are conflicting reports. Well, when we look at the genotype, there is AA, there is AS, some people are SS, some people are AC. Some people say that those who have AA do not get malaria very often. I beg to disagree, because I am AA, and there is no 2 months pass without me suffering for one form of malaria or the other. Mm-hmm. There are some sickle cell carriers, SS, who don't have malaria, and they are supposed to be the group of people that are most, qualified to have malaria. So there are conflicting reports. Mm-hmm. Some will say genotype has an effect, some will say they don't have an effect. Me, personally, I am AA, and I have malaria at least 5-6 times in a year.
Mm-hmm. Thank you very much.
Thank you, sir.
Just one last question back, please.
To add to that, you see, the AA, they are more prone to malaria because the plasmodium needs oxygen in order to survive. Now, those AA, they have a lot of oxygen in their blood. That's the hemoglobin. It's very effective, and then the plasmodium survive very well. But those with AS and SS, they have less hemoglobin, and then they rarely have plasmodium because there is no oxygen for the plasmodium to survive. Let's end it for another person.
Okay, one last question, please. Back.
Thank you for your good presentation. But if you eliminate or control malaria and mosquito in Nigeria, it will come from neighbor country. What about the programs surrounding area?
You see, this advocacy is not only for Nigeria, but I have to limit myself to Nigeria now. Because if Nigeria takes the bull by the horn and embarks on integrated vector management, and there's a significant reduction in morbidity and mortality, the whole of Africa will key in. The whole of Africa will key in, because 1 out of every 4 Africans is a Nigerian. 1 out of every 4 Africans is a Nigerian because of our huge population. So if we can do it and be successful with it, the whole of Africa will key in. And if the whole of Africa keys in, and there's significant reduction, not more than 2-3% for both morbidity and mortality, you can imagine the effect or the input of that into the economy. There wouldn't be any absenteeism again. People will not be sick. They will always be able to go to work. Their productivity will increase. The GOP will increase. So the advocacy actually is for the whole of Africa. But because I'm a Nigerian, let us start with Nigeria first and see. If you are successful, Africa will be successful in controlling malaria. Thank you.
Okay, thank you very much.
It's finished. Next one. Next speaker is Dr. Ukpai from Michael Okpara University of Agriculture, Nigeria.
Do you know which is your talk here? Is it nice? Is that one? Yeah. Oh, no, yeah. Is that it? Yeah, great. Okay. That's the pointer there.
Okay, that's the story.
Yeah, so that's
The scroll.
that's forward, backwards, and that's the pointer.
Okay. Thank you. Hello, everyone. I'm going to be talking on the prevalence of malaria, knowledge, attitude, and practices about malaria among attendees to the Medical Center of the Michael Okpara University of Agriculture, Umudike. Well, that has to be me. Introduction. What is malaria? The last speaker has talked about malaria. So we all know it's a mosquito-borne infectious disease of humans and other animals caused by the parasitic protozoa of the genus Plasmodium and transmitted from human to human through the bite of an infected female adult Anopheles mosquito. It is largely distributed throughout the warmer regions of the world. Malaria is wholly endemic in Nigeria, and more than 2 billion people are at risk all over the world, with over 200 million cases each year. Over 600,000 deaths occur each year, and malaria kills a vast number of children each year in Africa. In Africa, a child dies every 30 seconds from malaria, and that translates to a massive 2,880 children per day. I think that's a great mortality. Talking about prevention, he has dealt with that. Prevention may be more cost-effective than treatment in the long run. Initial costs required may be out of reach of many of the world's poorest people. The presence of malaria in an area requires the combination of high human population density, high Anopheles mosquito population density, and high rates of transmission from humans to mosquitoes and from mosquitoes to humans. So prevention could therefore be through medication, the intermittent preventive therapy for pregnant women, weekly preventive medication. In our time, we're giving Daraprim every Sunday, so I don't know what obtains now. And of course, mosquito elimination, which is the vector control, controlling both the adults and the larvae. We have to decrease availability of open water for breeding, and of course, add substances to decrease development. Prevention of mosquito bites is important through the use of repellents, insecticide-treated mosquito nets, and indoor residual spraying. ACTs are very important for treatment due to drug resistance syndrome. So what is the objective? We set out to determine the prevalence of malaria among those who attended the Moayu Medical Center and to determine the knowledge, attitude, and practices of the respondents regarding malaria. Mumdike is in Ikwano local government area within those coordinates. It's a tropical climate region, rainforest vegetation. And the population you find in Umudike are mainly students, civil servants, farmers, and petty traders. Before data was collected, of course, we had to seek permission from the authorities before we commenced our work. The patients we were about to work with, we had to inform them about the study, and only those who consented were included in the study. Questionnaires are distributed to gather socio-demographic information, and of course, the knowledge, attitude, and practices about malaria from the respondents. Venous blood samples were septically collected from both male and females of different ages using disposable sterile syringes. Thick and thin blood smears are made from the blood samples in grease-free slides. Field stains A and B were used to stain the thick blood films, while the thin blood films were stained with Leishman stain. Slides were examined for parasites under the microscope. From the respondents and the questions asked them, we discovered that the most common illness they reported was malaria, though there were other things reported like diarrhea, measles, cholera, but malaria was the most common illness reported, and of course it is not out of place considering the environment that we are. About 26.86% were infected. More males than females were also infected, and that's what we can find in that table there. Considering the age groups, we saw that the age group 61 to 70 had the highest rate of infection. Probably when we come to discussion, we'll be able to discover why. Knowledge, attitude, and practice studies. Many of the respondents had heard of malaria. 97.43% had suffered from malaria before. Fever was the highest symptom reported. Other symptoms included vomiting, body pains, headache. And so that's a table showing us the symptoms that were reported by the respondents, those who have heard of malaria and those who had suffered from malaria. So what were the causes of malaria that were reported? Some said mosquito bites, okay. Others said contaminated food, and some palm oil. So from this, we could see a case of ignorance being displayed. Even those who said mosquito bites couldn't even place the cause of malaria, because it is not just mosquito bites. So what was in that bite that caused the malaria? In fact, some few days back, before I came here, some newscasters were talking and they said malaria caused by mosquito. And I said, what? Why would they say that? Because Plasmodium causes malaria, not mosquito. Mosquito only helps to transmit the Plasmodium. So a good number of people don't even know that Plasmodium is responsible for malaria. So that's the table. And then there were treatment measures some of them took. Some visited the hospital, and that was about the highest percentage of treatment measure taken at 77.1%. Others went to the chemist, a case of self-medication. And still others went to the use of herbs. We have things like dogoyaro that people use for malaria. But the danger here is that the dosage is not known. For prevention, they still made use of malaria drugs. Some used insecticide, and some used mosquito nets. The 26.8% infection rate that was found in this study differed from the reports of Okpa and Ajoku 2001 and all the other authors we can find there. Some factors like climate factors, high temperature, rainfall, humidity, which favor breeding, could have contributed to the transmission of infection. Location and environment could also be a contributory factor to infection, as the respondents came from different communities around the university to seek for medical attention. The university is situated in a rural area, and as part of its community service, patients come to the medical center to seek for medical attention. Attempts at vector control will probably have differed, of course, at the different communities, leading to vector-human contact and infection at different levels. More males were infected than the females, though this differed from some works like Umaneto et al. 2006. Again, occupation could be a contributory factor here, as males work as night guards, or what we call security, and a good number of times they relax outside in the evenings with friends over bottles of drinks, what we call the stockfish dish, and of course, in doing this, they get themselves exposed to the bites of mosquitoes. In terms of age, we found that 61 to 70 years were the highest infected group. And we want to infer that because of the few number that came to the clinic, this could be why this is so. Many people in this age group are usually taken away by their children to the town to see a private doctor, or they will arrange for a home doctor to come and see their parents. So these ones that came probably were not privileged to have such arrangements made for them, so that could have accounted for why there were few, and those few had infections. The symptoms and signs reported were similar to those reported in other works. Fever, though, stands out. This is the most severe aspect because if it's not controlled, it could affect other workings of the system, as enzymes could be denatured at very high temperatures. Possible causes. Mosquito bites were the most mentioned, though they could not really say what was in the bite that led to the infection. Some others pointed to palm oil and contaminated food. In this case, health education becomes very imperative with the communities, and that means they have to be educated on the cause, the symptoms, the prevention and control of malaria. In conclusion, vector density and human population density are very vital to transmission of malaria. And so proper vector control should be implemented. The environment should be kept clean. Containers with water should be poured out. And there should be no water in the environment to provide breeding spots for the mosquitoes. Health facilities should be provided in the communities. wish to thank God for his love, mercy, and strength, and my students, all the respondents, and lab staff of Medical Center Mwayu. Thank you for your time.
Questions? No? Thank you very much.
Okay, that's the close of our session today. So I think we've had a really interesting day, some very diverse talks and really interesting talks. So I wonder if we could just have one last round of applause for all the speakers. If